Very good morning and welcome you all. Uh, First of all, I would like to welcome Dr. Asutosh here. He's our first presenter. Before starting today's program, I would like to conclude. I would like to introduce about yesterday what we discussed. So this is second day of this 5 days FDP, and first day we had in first half we had this inaugural session in which our mentor, our domain director, different coordinators, they gave their speeches and their views about this green technology. And after that we had three four eminent speakers, and they discussed about different aspects of green technology and how we can improve our environment that will helpful for the sustainable development. So now today is the second day of this FDP, and the theme for today's FDP program will be role of modern genomics tool in wildlife conservation to meet the goal of sustainable development. So today, whatever we will discuss, we have 5-6 speakers for today's. So they all will brief us how modern genetics tools will help us to conserve the wildlife. And if we will conserve the wildlife, ultimately we will conserve the forest, and that will be one of the best way for the sustainable development. So now genomics is very modern tool, and they have that this tool have enormous potential to conserve the species, to understand about the diversity of the species, how they are moving in this different national park, what is the demography of that particular species. All those things we can do without harming the animal, without harming the species, without harming the environment. So this tool is very promising tool. So hope whatever speakers we have, 5-6 speaker, all will give their best here, and that will be beneficial for the our faculties. So now we are starting this webinar, and I would like to invite our coordinator, Amit Institute of Forestry and Wildlife, Dr. Randeep Singh, to give their opening remarks. Randeep, sir.
Thank you, Dr. Sujit. Dear esteemed guests and participants, so namaste first of all. It is with a great pleasure I extend a warm welcome to all of you to our second day of FDP. As you know that we have started this FDP yesterday, and this event is conducted with the blessings of our founder president sir and support of our dynamic chancellor sir, Dr. Atul Kumar Chauhan sir, guidance of our honorable vice chancellor Dr. Balwinder Shukla, and our mentor Dr. D.K. Bandapayal sir. And this program is conducted with the help of MIT Academic Staff College and in association with Natural Resource and Environmental Science. So as Dr. Sujit says, the title of FDP, as we are familiar, building a sustainable future with the green technology, innovation, opportunity, and challenges. As you all are aware about the Sustainable Developmental Goal, and for your knowledge, I just brief you. Sustainable development is development that meets the need of the present without compromising the ability of future generations to meet our own needs. In 2015, the United Nations General Assembly adopted the Sustainable Developmental Goal from 2015 to 2030 and explained how the goals are integrated and indivisible to achieve sustainable goal at the global level. So as our title says, sustainable future. So UN Sustainable Goal 15 is life on the land. Like we are from the natural resource and environmental science domain. So our motto is the motto of this goal is 15 goal is to protect, restore, and promote sustainable use of the terrestrial ecosystem, sustainably manage forest, combat desertification, and halt the reserve land degradation and halt biodiversity loss. Just about the international biological diversity. So keep of that day was to bring the biodiversity back. So again, I extend a warm welcome to all of you to our event focusing on the role of modern genomics tool in wildlife conservation to meet the goal of the sustainable development. This occasion make an important step forward in our collective effort to safeguard these diverse and precious species that inhabit on our planet Earth. As we stand at the cross of the progress and the environmental responsibility, it has become increasingly clear that conservation of the wildlife is not just a modern moral obligation, but a critical component of the sustainable development. The, the integrate web of the life on the earth, which it is countless interdependencies. For example, the ecosystem services, our dependency of the natural resources, and delicate balance hold immense value for our ecosystem and our further generation. In this context, the emergence of modern genomics tools has revolutionized our approach to wildlife conservation. As you are aware that India is a country of the rich and the biodiversity. So the biodiversity is have the immense pressure of the anthropogenic pressures, and lot of these pressures are coming on the biodiversity. And human breeds, it's basically depleting our ecosystem. So lot of poaching of the wildlife happens, lot of smuggling, lot of killing of the wildlife happens. And there are the lot of opportunities we have to save this biodiversity using these modern tools. So genomics with its ability to unravel the intrinsic DNA code of the species, like we all are the unique DNAs, it's offer unparalleled insight into their biology, genetic diversity, and adaptation strategy. By hashing this knowledge, we can make informed decisions, diverse, effective conservation strategies, and ultimately work towards the preservation or the conservation of the biodiversity. The utilizations of genomic tools in wildlife conservation endangered species, yet lot of species they have been discovered. So our there are the scientists who's joining shortly in 2 days from the JDSI. They have discovered recently macaca species, which basically in the northeast part of India, and they have resolved some of the ambiguity. And gaining a understanding of the impact of the environmental change on the wildlife. On this knowledge, we can implement target intervention, develop robust conservation plan, and mitigate the threats and jeopardize the survival of our cheers wildlife. So I would like to express my deepest gratitude to our distinguished speakers and our panelists who have graciously agreed to share their expertise and insights. So the speakers, your contributions will be undoubtedly inspire and shape future of the wildlife conservation. I also extend my heartfelt thanks to all my participants for joining us in this crucial endeavor. May this event be a catalyst for transformative actions and innovation, raising awareness and fostering a deep appreciation for the invaluable role of genomics in wildlife conservation. Together, let's strive to create a sustainable future where humans and wildlife can coexist in harmony. Thank you and enjoy the event. Now I'll ask Dr. Sujit to introduce the speakers and continue this.
Thank you, sir. Thank you for your opening remarks. Before starting talk of Dr. Ashutosh, I will take little more time because I want to introduce about all the speakers little bit. Then I will invite Dr. Ashutosh. So I'm sharing my screen here, and so I think it is visible to you all. Yeah. So as I discussed earlier that this is the theme for today's talk, and today we have 6 different speakers. So our first speaker is Dr. Ashutosh, and currently he is working as a scientist C at Southern Regional Center of Wildlife Institute of India, which is situated in Coimbatore. Initially, this institute was known as a SECON, that was Salim Ali Research Institute for Ornithology. And he will talk about glaciation and climate change impact on birds and how genetics will help us to understand these effects. I will be the next speaker, and I will discuss mostly about what are the tools are available to understand the to conserve the wildlife, especially genetics and genomics tools. And our third speaker will be Professor Joni Aspi, and he is professor in University of Oulu, Finland. He is also director of the Department of Ecology and Genetics. And he will mostly talk about level of inbreeding and inbreeding depression among Finno-Scandian wool, which is distributed in Finland, Norway, Sweden, some part of Russia, and Estonia. Our next fourth speaker will be Dr. Puneet Pandey. So currently he is working as a senior research scientist at Seoul National University, South Korea, and he will mostly talk about protecting wildlife through technical scientific cooperation. He will mostly talk about illegal trade and how we will apply genetic tool to understand all these things. Our fifth speaker will be Dr. Rita Sharma. She is research affiliate in Wildlife Staff India, but mostly I can say she is among the very pioneer scientist who started genetics work in India, and she was among the few I think first or second PhD students who applied genetics tool for tiger conservation in India, and she was Dart Fellow, and she did her PhD from Potsdam University, Germany, and after that she spent lots of time in Portugal for her postdoc, and she worked on especially conservation of elephants. Recently, she is also working with affiliation with Wildlife Institute of India for the conservation of elephant. So she will talk about how genome-wide single nucleotide polymorphism markers are helpful for the conservation of the endangered Borneo elephants. And our last speaker will be Dr. Bhimdat Joshi. He is scientist, Zoological Survey of India. And he will mostly talk about conservation genomics of endangered mammals in the Indian Himalayan region. So it was very, I can say important project run by the ministry, and JDSI did one wonderful job, and Dr. Joshi was one of the key part in that project. So he will talk about all these things, how he will apply genetics and genomics tool for endangered species in Indian Himalayas. So this was some brief introduction of our all the speakers. Now I would like to invite Dr. Asutosh for his presentation, but before his presentation, I would like to introduce a little bit about Dr. Asutosh. So I can say that Asutosh is alumni of MIT, and he did his MTech from MIT University in Biotechnology. And after that, he joined Gurukul Kangri University for his PhD and Wildlife Institute of India. So he mostly worked on the bird speciation, and he is one of the student of best ornithologist, I can say, Dr. Trevor Price in Chicago University. And Dr. Asutosh in last, I think, 10 to 12 years he spent, and he did tremendous job for the wildlife conservation, especially bird. And currently he's serving as a scientist C at now Wildlife Staff India Southern Regional Center, and he's looking all the wildlife forensic and genomics and genetic facility there. And he's working for conservation and genetic monitoring of different bird species in different parts of India. So I would like to invite Dr. Asutosh to deliver his lecture. Dr. Asutosh, please share your screen. And
thank you so much for the nice introduction. And being an alumni of the MIT, it is always privileged to deliver even some lecture which actually I can contribute and encourage the students for conservation in the wildlife. So thank you so much. Is it my slides are visible?
Visible, Dr. Asutosh.
Okay, sir. So already you have introduced, so there is nothing to introduce myself. So I will directly move on my topic. Actually, already you have told that I will talk about the glaciation and the impact of climate change on the Himalayan bird species. So in this, I will actually put more emphasis that how the this physical barriers and the variance has resulted into the evolution of the various bird species in the Himalayas. But every every coin is having the two sides. One is we have the positive side, another is the negative. So the way in Himalayas, many bird species has evolved, but on the other side, it has also restricted for the new species to come up, because already most of the space are occupied by the existing bird species. So I will tell about that how the different species has evolved and what are the current threats and how the niche partitioning actually works and the contribution of the citizen science. Also, along with the modern genetic tools that we have applied to identify the different species in the Himalayas. So coming to my presentation, it is a well-known fact, and most we all are actually aware that Himalaya is one of the major biodiversity hotspots, and it is also known as one of the youngest mountain series because it has evolved only during the last 6 to 8 million years ago itself. And still, it is believed that the Himalayas is still rising 1 centimeter every year. And due to its extensional elevation gradient from 150 meter up to 8,800 meters, it is the region is actually very rich in floral and faunal diversity. But what we see about the Himalaya was not like that. When the Indian plateau has collided with the mainland Eurasia, at that time, immediately after that collision, the Earth has gone under the major glaciation. And there are the several theories which states about the glaciation period. One of the stories says that during the summertime, this northern hemisphere was not able to receive the sunlight due to its orbital position and its tilting, due to which the most of the time the Earth was not receiving the sunlight for the thousands of years, due to which the Earth's northern hemisphere became very cold and the seven layers of the snows has get accumulated, due to which the ice age was triggered. Whereas the another theory which the most of the astronauts say about the glaciation is that one of the big asteroid has collided with the Earth, due to which the a lot of the dust has surrounded the Earth atmosphere, due to which the sunlight was not able to come up to the Earth's surface and the temperature has become down, and due to which the Earth has underwent into the glaciation period. Due to the various tectonic events and the snow, just like I have told that still the Himalayas is rising, the several vicarians and the various barriers has been formed in the Himalayas. And due to the tectonic events followed by the glaciation, and ice age was formed. Ice age formed the several valleys and ice-capped mountains. Further, it was also believed that after the last major glaciation, since the eastern Himalayas was very close to the equator, so it was very warm and the humid, and the evolution of the species started at the eastern Himalaya. At that time, most western Himalaya was very dry and devoid. None of the species were present. But as the species has evolved and the western Himalaya started getting warmer, some of the species migrated from East Himalaya to West Himalaya. But they were not able to come back due to the lot of the vicarians, mountains, and the rivers. So it has actually resulted into the development of the new species. Just like here, it is an example of the how the mountain tops act as a barrier, whereas how the valleys can act as a barrier. In this image, you can see that if one species is having a wide range of distribution, when it is when the species get isolated by the physical isolation, later over the period of time, due to the reproductive isolation and the change in the genetic structure, they will evolve as a new species. For the species which are the ground dwellers, they are also for them the valleys act as a barrier, and later on, due to the physical isolation and the reproductive isolation, they will evolve as a new species. In this image, you can see that how the ice can actually help. In the first image, you can see how the ice-capped mountains can act as a way for the movement of the species which are alpine, means arboreal in nature, or those who are residing at the top of the mountain. So they can migrate from one place to another. But when this ice will get melt, then they are actually not able to come down up to the valley, and they can actually go to the next they can actually cooperate with their existing one. That will actually result into the reproductive isolation. And I don't have to go about the detail about what kind of a different kind of speciation that actually the birds have. So these are the actually type of the speciation that birds follow. We will just come into the type of the speciation. Just like we always say that in Hindi we call the murgi aya anda. Just like that, these tragopans, which are actually belonging to the hen family, they are actually evolved around 2 to 4 million years ago. What happened during this time? During this allopathic speciation, Tragopan in two different episodes about 4 and 2 million years ago. In those periods, the vicariant events that might have triggered the allopathic speciation in the bird. Just like here, you can see around 4 million years ago, Technic Tragopan and the Kobaki Tragopan has just evolved as a new species, whereas 2 million years ago, the Western Tragopan, Satire Tragopan, and the Blight Tragopan, they have also been evolved as a different species during the same time. So, which is actually representing that they have been speciated into the different species based on the physical isolation, but they are actually following the allopatric speciation. Then there is a one perfect example of is the alpine thrush. Actually, earlier the alpine thrush was known to occur from the Ladakh up to the China region, but later on in 2016, Alstrom et al. Have segregated them based on their genetic, vocal, and the morphological characteristics. Just like in this image, you can see that this alpine thrush is having the two species, means the two subspecies. Actually, it was earlier considered as a single species, but later on, when they have carefully examined, they found that the species, there are the two individuals which are having the different morphology. The one individual is having the very prominent black strip on their wing, whereas the other one is having very the thin strip. Further, when they noticed, the nominate form was having the yellowish leg, whereas the second the individual which they have observed, it was having the pale leg. Further, when they looked up into the breast part, you can see that the above species is having the more yellowish or the rufous breast, whereas the Himalayan thrush is only having the pale breast kind of a thing. And lastly, when you will look up into the mustaches, then you can see, if I am able to point it out, that you can see that this mustachial strip was very prominent in the alpine thrush, where it is very narrow in the Himalayan thrush. But how this got has happened, and because they were not actually in the field, it is very impossible to actually identify, because if sometimes the sub-juvenile are also having the same features like the male nominate form. But this has happened only because of the genetic studies. Considering the same thing, we have also conducted one study and taken Cyanus rubiculoides complex as one of the model species. For that, before coming into the analysis part, I will tell you some little history about the rubiculoides. Here, Cyanus rubiculoides, or you call the blue-throated blue flycatcher, were having the 5 subspecies, among which only the 2 subspecies, Cyanus rubiculoides and the Dilemmus, were known to occur in the Himalayas. Whereas the 3 species, the Glochiclomus, Cyanus helenus, and the Clothi, they were actually present in the different. So Zhang et al. In 2014, they published a paper and they identified that which Cyanus rubiculoides glochicomus, which was considered as a subspecies of the rubiculoides, is a completely a different species, and they named that as a Cyanus glochicomus. The subspecies Clothi, they have merged with the Cyanus helenus, and it is based because of their genetic structure. Then what I did for my study, I have collected around 22 samples, which are actually ranging from different elevations from east to west Himalaya. So I have collected the samples from 300 up to the 1500 meter elevation. And when I just, and because this species was also considered as a single species throughout the Himalaya, I made a genetic analysis. What I found that the species which were present at the higher elevation, they are forming a totally different plate, whereas those species which are present in the western Himalaya, they were forming a totally different plate. And you can see with the blue one, the eastern Himalayan high elevation one is with the individuals which I then at the lower elevation, 200 to 300 meters, which is represented by the green one, you can see they are actually forming a different tree, which has given us a clue that they could be a different. And when we run a time calibrated phylogenetic, found the individuals which are segregated means at the higher elevation, they are separated by the 1.52 millimeter rule, whereas the individuals which are only present at the foothills of the Himalayas or the lower base, they are segregated by the 2.5 millimeter rule. Speciation concept. If any species which is segregated in the 2 million year rule, they should be considered as so. When we made an analysis, we found that there is no actually genetic inflow between the there is no genetic data transfer between these problems. Means they are actually not dealing with each other. They are completely isolated. Even just like I told you, the eastern Himalayan lower elevation population and the higher elevation population, both are actually present in the same region, but at a different elevation, but they are separated by the 3 million years. So what I have told you that how the mountain barriers and how the mountain and the valleys can act as a barrier in the physical isolation of the species. This is the perfect example, and this is also an example of the parapatric mode of the speciation in the Himalayas. And you can also see that here, if you look into the morphology of the species, then you can see that those species which are at the higher elevation population of the eastern Himalaya is also having the round throat like the western one. But the one which is got completely segregated, it has evolved a different morphological feature, and the throat is like the throat is actually forming a notch, which is actually the V shape. So which later on after the genetic study, we identified this feature also. And when we put our data with the Zhang data, who has already segregated the Glaucicomens and the Clossi, then what we found that the lower elevation population is actually not a subspecies of Rubiculoides, but it is a subspecies of the Hyanenus, which is actually a Chinese species. So which has based on that, we have revised the taxonomy and the distribution of the species throughout the Himalaya and the Southeast Asia. So based on that, actually the conservation implications can be actually taken for the effective management and the conservation of the species. So if I talk about the adaptive radiation, so above all the studies are indicating for the adaptive radiation that species acquired and diverged into different species. They all have developed different morphological characteristics up to small or large extent. Just like this is an example of the finches which are present in the Himalaya. Here you can see that how this adaptive radiation has resulted into the evolution of different species. But this adaptive radiation and speciation has resulted into the evolution of many species that in turn resulted into the niche filling and leaving no new species for any species to come up. If any species has to come up, then if any force led them to evolve, they must have to replace any existing species. Like the same way, Trevor Price has represented that species distributed across space which has become fragmented due to the vicariance or the disperser of the reproductive isolation that has taken over all the available space, leaving only the little space for any new species to come up and evolve as a new species. This is the actual time. Now we are coming into the how this Holocene temperature has impacted on the means extinction of the species, and how the genetic tools will actually help to help us to actually prepare a proper genetic conservation plan. So when if we see about our Holocene, which actually starts from the 12 million years ago, you can see here that the rise in the greenhouse gases was initiated around 8 million, around 7 million year ago, and now the temperature of the Earth is around 2.5 degree warmer than what it was after the glaciation period. Also, Sabine has also shown that during the last 7 decades, the Earth's surface temperature and the Himalayan temperature, when they have compared, they have found that the Himalayan temperature is actually rising more rapidly than the Earth average temperature, which is actually when we will compare the Himalayas. Now with this image, you can actually compare that what will be the future of our Himalayas. That means whenever there will be a ice, means it will, due to the high temperature, it will get melted down, and later on we will only have the runoffs whenever the rain will come. And Sabine et al. Has also shown that during the winters in the future, there will be a very the surface temperature will rise in a very rapid rate, and the annual precipitation will be actually a short. The precipitation will be short but rapid. So that will actually result into the flood conditions, and there will be a sharp decline in the snow days, which actually represent that at a one time you will have the enough water, but at certain time you will not get the water for that. This is a study which we have conducted at the Bhimgada Barrage, Haridwar. This is actually a barrage which is actually receiving a water from the river Ganga. And at the Bhimgada Barrage, at the center of the barrage, there is a island which is actually having a lot of the vegetation, which is supporting a lot of the migratory bird. And most of the whatever the birds they are coming, they are actually the diving or the dabbling nature of them. So in Haridwar, the 3 major floods has happened in the past, which happened in 2007, 2010, and the one of the well-known flood was the 2013 Kedarnath flood. Then with this image, actually you can compare that in 2003, the first image on the right left-hand side, you can see that there is a very big island was there, and it was covered with the vegetation. Just the figure 1b, you can see after the 2007 flood, there is a gap which is evolved due to the flood conditions. Later on in 2010, you can see the another flood came, and it also just eroded the vegetated land. And after the 2013 flood, you can see that only the small patch of the vegetated land is available for this migratory bird. And this was the condition of that barrage immediately after the 2013 flood, which represent that there is only very limited space is there for any species to come and survive there. And this is actually the population trend. You can see, just like I said, the Haridwar received the flood in 2007, 10, and 13. You can see here that how the population of the migratory birds have declined drastically when they were coming to the Haridwar. Actually, these flood conditions are not only impacting the migrating of the bird, means it doesn't represent that they will stop the migration. They migrate, but they actually delay their migration. And migration is not a delay in the migration due to the climate change is not only in the birds, it is also observed in many other species. One of the very good study which has been conducted by the Hayes et al., who has actually used the different climatic variables to map whether which factor is actually responsible for the delaying the bird species migration. When he has taken over the various factors, he found that the temperature is one of the key factors which are actually responsible for delaying of the bird species migration. Further, this is the finding of one of my supervisor, Dr. Trevor Price, who is actually conducting a long-term data from the uh, Western Himalaya, from the Kashmir and Himachal Pradesh. So here you can see that, sorry for the image is not clear, but in the first image you can see that it is actually the mean spring temperature on the y-axis, and the x-axis is the year. Here you can see that the temperature in the 1985, with the average temperature was around 15.2 degrees centigrade, whereas in the 2010 the average temperature was something like 16.7. Means over the period of something like 2 decades, the temperature of the Western Himalaya has risen by the 1.5 degrees centigrade. When he has actually compared the data with the breeding time of the birds, then you can see in 1985, the birds, mostly the warblers, they were actually breeding in the first week of the June. In the second graph, it is a mean breeding date from the 1st January. And you can see in 1985, the birds were actually breeding on the 185th day of the year. Whereas due to the change in the climate condition and becoming more warmer at the later phase, in 2010 you can see they are actually starting breeding in 170th day of the year. Means they have actually shifted their breeding 2 weeks earlier, which is also having an impact on the other bird. This is one of the perfect example of the shift in the migration. This actually the study was conducted by the Sampolonius and the both, which have compared the arrival and departure time of the flycatcher and the tits. The above temperature, the above graph, which shows that when the flycatcher and tit have the overlapping mm, breeding time at the same area, that will result into the fatal accidents. Means the fatal fighting, and it will result in the mortality. Whereas at the lower means at the bottom graph, when you see when there is a complete segregation in the breeding time of the flycatcher and the tit, means there will be a successful breeding for both species. Means the resource availability will be there, the other space just like the nesting sites will be available, so that they will not fight with each other. Secondly, that was the case of the space. Secondly, this is the another example when the climate the climate change will happen and the temperature will rise. What will happen? The species which are present at the lower elevation, just like the 500-meter elevation, what will happen? They will actually migrate to the higher elevation to cope up with their physical needs. So what will happen when the out of the four, when they will migrate to the higher elevation, they will definitely replace the species which are present at the higher elevation. And the species which are present at the mid elevations, they will move more further and they will force the other species to move more higher on the mountains. But the species which are already present at the higher elevations, they will not have any opportunity to go anywhere. That will result into the extinction of those species. Similarly, we have conducted one study on the Indian peafowl. Actually, we have got one image using the camera trap in the Darjeeling around 2,200-meter elevation, which has actually triggered our mind, and we just looked up into the distribution pattern of the Indian peafowl. And we found that the Indian peafowl is having the distribution only up to the 1,500-meter elevation. But when we recorded at about 2,200-meter elevation, we got surprised and we started gathering the data from using the citizen science data. What we found that after the 2010, there is a frequent record of the species, frequent sighting of this species above the 2,000-meter elevation. Further, when we try to investigate whether the species is means all right, means already they have migrated to the higher elevation, we found that no, these species are actually during the summer, they are more frequently visiting on the higher elevation compared to the winters. That represent during the summers, they are actually not finding an appropriate place for their breeding and their survivability. This is also the study from the Western Ghat of the Indian peafowl, where Josh and Namir has found that there is a very limited space for the Indian peafowl present in the Himalaya, in the Western Ghat, which is at the center of the Kerala. But when they have actually done the modeling for 2050 and 70, then they found that the distribution range of the species will increase up to 41% and 53% by the 2050 and the 2070. Whereas their habitat will be actually declined by 22 and 32% by the 2070, which is actually representing that the habitat which is now suitable for these species will get totally diminished due to the climate change. And it is also found by the Reno et al. That if there is a 1 degree rise in the temperature, that will cause the risk extinction, the invasion by the 47%. Mean the 47% of the species will definitely try to move higher on the mountains. And that will also result in the extinction of the species. Already we have lost 2 species, that is the Himalayan quail and the pink-headed duck. And actually, the actual cause of this their extinction is not known, but it is also relating with the climate change. And as per the data, over the period of the 500 years, we have lost something like 190 species, which is contributing around 1.5% of the total existing species of the world. And as per the current studies, there are around 1,500 species around the globe which are actually facing the extinction due to the climate change. And basically half of the known bird species are grappling with the population decline. And this is the current scenario of the bird species which are actually facing the threats. And if you will see the critically endangered species, in 1994 we had only 2 critically endangered birds, but over the period of the 2 decades we are having the 17 species, which is representing 850% increase in the critically endangered bird species in India itself. So, and if I talk about these, the critically endangered bird and the threatened bird, most of them are actually the migratory in nature. And in India, the most of the migratory birds, they actually arrive in India through the 2 flyways. That was the Central Asian flyway, and second one is the East Africa West Asia flyway. But however, when they enter into the India, they start facing the threats, just like they are actually chased by the dogs, the pollution, and the capturing by the nomads. This is only that we are not actually facing the threats. We also have the conservation examples as well. One of the best conservation example of the migratory bird is the one of Amur falcon. Actually, this Amur falcon is a bird which is actually native to the Amur region, which is situated just below the Russia and next right side of the Mongolia. This bird is known to actually track the summers. So during the September October, when the Russia starts getting cooler, this bird start migrating, and they usually migrate up to the next side of the equator, which is actually become warmer during that time. So during the month of the October and November, they actually come and congregate in the northeastern part of the India in the million number of the time. And earlier, the people were actually using and killing them for their as a cheap source of meat. But later on, due to with the help of the citizen science and the people contribution and the forest department contribution, the 3 birds were actually radio tagged. That was the Naga, Pankti, and Vokha, which were named after the Nagaland state, Vokha district, and Pankti village. And people were actually get interested into the conservation of the bird. When this bird was got tagged, because this bird actually take an annual migration from Amur region to the South Africa, then again they will come back during the summers to the Russia. So these people actually got interested and they started conserving the Amur falcons. And later on, what happened, due to the availability of the citizen science data, you can see that they have tracked their movement
throughout the year. And you can see that during the October, November, they are mostly coming to the India part. Then they are going in the December in the South Africa. Then later on again they are coming back to the Amur region. So that is not only the one example. Around the globe also, you can see that the Cornell lab has actually tracked the migration of around 118 species. That represent that at what time they are actually present in which region, which is actually helping to prepare a conservation plan at an appropriate time, so that the data and the samples could be collected. So with this, actually, I will just put some emphasis that we still have the gaps in our knowledge. Britishers have started the bird watching in India, but what happened? So later on, it has paused for several times, but later on again it has taken a complete drastic change, and now again the citizens have contributing towards the citizen science. So the geographical distribution of the birds we only know very limited based on the regional level. So the migratory routes and the seasonality of the species could be only known with the help of the not only with the scientific data but also with the citizen science data. And impact of this changing human modified environment need to be actually reduced, and the conservation action required, they should be actually based on the genetic data as well as their distribution. So it is actually required to have the complete diversity portal for completely diverse diversity. Unless we are not aware about the complete diversity of the species, we are actually not able to plan any conservation actions. And these are actually possible through the modern genetic tools only. So with this, I will say, I will stop my presentation. Thank you. And I would like to thank to the ministry for giving an opportunity to conduct the field works, Director Secon and WI, Dr. Randeep sir, Associate Professor, and Dr. Sujit sir for inviting me on this platform.
Thank you, Dr. Asutosh. Thank you very much for your talk. Now, Asutosh, I have one question, especially. Suppose you are looking different altitudinal gradient in the Himalayas. So obviously, if you see literature in Himalayas, if altitude will change, there are change in species distribution and species diversity also. So can you explain how it will work, how genetics will help us to detect these things?
Actually, just like I just said that the species actually requires favorable conditions to evolve. And at the lower elevation, there is a less diversity. The region which is having the more diversity, just like the floral diversity, it will have the different diversity of the flora there. That will support the faunal diversity itself. So if we talk about the modern genetic tools, the eDNA, which actually we get the information about the diversity. With the help of that, actually we can identify the diversity of the species which are present in that region. So with the help of that, we actually we can plan for the conservation actions.
Okay, Dr. Asutosh, thank you for enlightening about the distribution of different species in Himalayas and how climate change affect their distribution. Thank you for giving our nice talk. So now we will move for next talk, and I will be the next presenter. So I will basically whatever Dr. Asutosh presented here, he discussed here different cases, how species are changing, how they require different kind of habitat, how they are interacting with different kind of climate change, and how genetics will help us to understand all these effects. Because understanding effect of climate change or any kind of habitat driven change in the species, it is very tough to understand with the help of normal ecological tools. So obviously genetics is one of the most promising tool to understand all these things. So now for the participant, I'm going to give a brief talk about what are the genetic tools are available basically. If you have these kinds of problem, whatever Dr. Asutosh have discussed here. So how you will apply these tools, how genetics tools will help us to understand all these things. So I will discuss very brief and very basics thing about the different tools of genetics and genomics. So I would like to start my talk here. So nice. So my slides are visible, Randeep sir?
Huh.
Yeah.
It's fine.
So very good morning, everyone. So I'm Dr. Sujit Kumar Singh. I am working as an assistant professor in Emitune staff forest and wildlife. This is part of this natural resource domain. And I will discuss about a tool in wildlife conservation, especially genetics and genomics tool. So whatever Dr. Asutosh, what kind of question he raised, how different climatic change and different habitat behavior will affect the species distribution and their survival. So I will discuss about how we will apply genetic tool and how we will understand these effect, because generally it is very tough to understand with the help of normal ecological tools. So if you see threat for the whole biodiversity, I can say, or we can say species, so which will lead to the extinction. So there are two type of threats. One is deterministic, another is stochastic effect. So deterministic threats are basically anthropogenic activities, which include habitat destructions, pollution, overexploitation of species, especially poaching, another thing, species translocation, especially type of invasion of species, and global climate change, because if climate will change, obviously that will affect the physiology of that particular species. And if species will be able to change their genetic makeup and physiological process, then they will survive, otherwise they will not survive. But there are many stochastic processes also there, where when any random change will be there, sudden change will be there, like genetic change, demographic change, and environmental factors, like many calamities, tsunamis, earthquake. These things will rapidly kill the species, and that will lead to the extinction of species. So understanding all these things are very important. So now the main thing is how genetics will help us, how these anthropogenic activity and random changes, how those things will affect the genetics, and how genetics will help us. So if any kind of issue or we can say problems, if you'll see what kind of major problem will arise if these two threats will work, your deterministic threats and stochastic threats. If these threats will work, then these problems will arise, and these problems will definitely affect the species survival, especially wild species, free ranging species. So first thing is fragmentation of population, and we know we have if you see the population of whole world, it is increasing day by day, and our natural resources are decreasing. So that will leads to the fragmentation of different natural habitat, and that will affect the movement pattern of species. And if species will not move, there will be problem in the genetic exchange, and they will start making one genetic pool, or population will be isolated. They will start breeding between the within their family, and they will go towards inbreeding or inbreeding depression. So if population will be fragmented, that will be small, and then they will start inbreeding, and that will lead to the inbreeding depression. Whatever term we are using here, these terms, our coming speaker will discuss about these terms. And if inbreeding depression will be there, there will be accumulation of deleterious alleles, which is not good for the population, or we can say harmful alleles will accumulate, and that will lead to the extinction of any population. And if you talk about captivity, if any animal is in captivity, definitely they will lose their wilderness. And if you have information of genetics, then we can help us to reintroduction program. Another thing is taxonomic uncertainty. If these factors will work, taxonomic uncertainty is very problematic thing. understanding how many species, different group of species is present, that is very important, just because of management. If we have how many different type of species are present, what are their habitat, what kind of habitat they are preferring, what kind of food they need, what kind of resources they need, then we can easily give them, those things, and we can manage them. So understanding taxonomic things is also very important. And if these threats will work, taxonomic uncertainty will be there, and we can lose our species. So these are the major threats. If we see as an effect of these threats, so these are the major issues we have, and all these issues we can discuss with the help of genetics. So I just want to little bit give you very simple example. You can see here, this is one very nice landscape. You can see here there is no barrier is there, I can say, but if you see this thing, yes. So if you whole population, there is no barrier, all populations are same, it means that kind of population can be called as panmictic population. So if there is no barrier, animals are free to move, they are free for random mating, then that kind of population can be called as panmixia. And these kinds of populations are homogeneous population, and generally in ideal condition, this kind of population is not exist. But yes, if there is no barrier, animals are free to move everywhere, then such kind of population can be called as panmixia. But suppose any barrier will come in the form of any kind of stochastic activity or any kind of your anthropogenic threat. If this threat will work, then what will happen? Population will start fragmenting. So you can see here, if this factor will work, population will be divided in the two different subpopulations. So obviously, if two population is subdivided, then anyhow that will restrict the movement of individual between the population. Now, whatever two packets we are looking, animals are bound to move in this area and bound to breed within family. Suppose if such kind of threat is more rigorous, then what will happen? There are many different pockets will arise, and whatever panmictic population we were looking, now these panmictic population is in fragmented, or you can isolated population. So each and every pocket have their own genetic makeup, they have their own survival, they have their own resources, and population is very small. And if population is very small, they are bound to breed with a relative, and if breeding in relative is going on for a long time, there will be inbreeding. Only few characters will go pass on in the next generation, and that will reduce the genetic diversity of the population. And if genetic diversity will lose, obviously that will lead to the survival potential of that particular species. If any kind of change will happen, the species will be unable to survive in that condition, and ultimately that leads to the extinction. So I just want to convey my message here. If there is no threat, what we have discussed, stochastic threat or deterministic threat, if these threats are not available, population will be in very good condition, healthy condition, panmictic condition, they are free to move. But if these threats will start working, population will be in this kind of pockets. So understanding these things, populations are panmictic or not, population are fragmented or not, this is very important for the management purpose. And identification of these things with the help of other tools, this is very tough. So genetics will only help us here. And if you see, if population is very small, so their resistance will decrease. So you can see here, if you see the generation time of population S1, S4, and S2, they are very small population, their resistance is low. And if you see the large population, their resistance is high. So if population is small, then inbreeding will be there, their resistance will decrease, and that will lead to the extinction. If population is very large, there will be less inbreeding, or we can say no inbreeding, and population will survive. So ultimately, resistance means what? Basically, that is their genetic makeup, how diverse they are. If they have more diversity, if their genetic makeup is more diverse, they will be more resistance. And if their genetic makeup is less diverse, there will be less resistance. So this is already proved by different studies on wild animal also. And if you see here, if population is inbred, then extinction rate will be higher. So you can see here, if heterozygosity is lower, means genetic diversity is lower, extinction rate is very high. And if genetic diversity is higher, extinction rate is very low. So if population is fragmented, if these threats are working, then population will go for inbreeding depression, and they will reduce their resistance, they will lose their genetic diversity, and their extinction possibility for the extinction will be high. So understanding genetics is very important for any species if you want to make a proper management plan, and those things we are discussing here. So ultimately, if you see whatever we have discussed, two things are very important. One is phylogenetic diversity, that is how many different species are present, how many different subspecies are present, how many different populations are present. That is very important. If you have idea about all these things, that will be very helpful for the management. And it is also very important how many population is present, how many species is present, and in what type of ecosystem they are living. So all these things we need to conserve, and we can conserve these things with the help of, ultimately with the help of genetics, and that will meet the goal of, species restoration, species conservation, and that will lead to the sustainable development. So generally, whatever we have discussed, this is very common thing for other kind of species, but if you are talking about wild species, it is very tough. And if you are going to use invasive method, which is not permissible for the wild animal, that is very problematic. If you want to radio collar one tiger, that is very tough task. It will take at least 6 months, a year, lots of human resources, lots of money required to, radio collar one tiger and collect blood sample or any kind of biological samples. And that will be very problematic for animal also. You can see this tiger have, large radio collar. So that will be very, problematic for them. If they will move in the forest, that will be not good for them. And this is not permissible also in many times. So radio collaring is one of the tough tasks, or we can say invasive sampling of wildlife is tough task, and this is not permissible. So I just want to tell you, in wildlife field, collection of biological sample is very tough task, and getting biological sample is really tough. And if you do not have good sample, then it is very tough to work in the genetics. So many studies have been conducted since last 1990s. People did lots of R&D, and now we have very well-established technique, that is non-invasive sampling, where we are collecting, indirect evidences of animal. So some technique are track plot. This is just, what we can say non-invasive science. And here we are not harming the animal, and we are getting the data that animal presence or absence. Sometimes we can see the camera trap that will also give you photographs, and you can identify the species. But for genetics, we need some samples, some biological sample. So for that, we need to collect the scat samples. Scat means basically fecal sample of animal. If you're talking about tiger or any kinds of felid, that is scat sample. If you're talking about other animal, there are different names like pellets for the herbivores, dropping, many other names are there. But generally, this is the fecal sample of animal. And this is one of the best source for getting DNA in the wildlife. Otherwise, you cannot get the DNA. So whenever we are going in the field, we are, collecting these samples, scat samples, and we are coming in the laboratory, and we are starting work, for the DNA isolation, PCR, and other things. So this is the best method. This is the one of the most important, most promising non-invasive sampling method, where we are not harming the animal, and we are getting the sample. And once we'll get the fecal sample, that will have DNA. We are assuming that will have DNA, because whenever fecal matter passing through the alimentary canal, they will have some mucus inside that. So we are utilizing those mucus, because little bit mucus as with scat sample or fecal samples. So we are taking upper layer of fecal sample and extracting the DNA. And there are possibility for getting DNA, but DNA is not very good quality. But yes, we are getting DNA, and nowadays we have different tools to work with small quantity of DNA. So if you have very small quantity of DNA, if you have very shear DNA, fragmented DNA, then you can easily do it with the help of non-invasive genetic techniques. So now sampling is clear. In the wild, there is no liberty to catch the animal, to take the blood sample, to take the tissue sample. This is not possible. And this is not permissible also under Wildlife Protection Act. So only one way is present, that is non-invasive sampling, where we will collect the fecal sample, and that will be our best resource for getting the DNA. So these kinds of non-invasive samples we can collect whenever we will go in the field. We can get hair samples. So if hair have some hair follicle, that will be enough for us. We can do something. We can extract DNA. We can extract the feces, that is fecal sample. We can also get DNA from the urine. If any bird is there, then we can use egg cells. If hatched egg cells are there, some some remains, tissue remains, or some amniotic fluid, these remains are there, so we can easily extract the DNA out of that, and we can do it. If any bone samples we can get in national park or wherever we are doing our work, then we can also extract DNA out of that. Any carcass we are getting, any dead animal is getting, we can get DNA. If said feathers we are getting, we can get the DNA. If scales we are getting, like pangolins have scales, so if you are getting a scale, we can get the DNA. Said skins are there, we can get the DNA. And if you are getting some saliva somewhere, we can also get the DNA. These are our potential resource to get the DNA from wild animal. Without harming the animal, we can get the DNA with these kinds of biological material. And if you are visiting any forested area, any one of these things you can get. So that will make your job easy to understand all those things, whatever threats we have discussed, what are the genetic diversity, what are the taxonomic uncertainty. All these things we will discuss once we will get any one of these samples, and we'll get DNA. So these samples are very precious. Whenever being a wildlife researcher, whenever you are going in field, try to see these samples. If you are getting these samples, collect these samples with the permission of forest department. That will be very important things for you. You can do many more things. You can tell about different hidden story of any forest, about any species. So obviously, once you'll get the biological sample, then we will leave the forest area, then we'll come back to laboratory and we will start our work. Now we will completely do our biotechnology work, or we can say genetics work. Now all these things, it's completely dependent on the DNA. And you all know DNA, genome, nucleotides. I think you all have idea about all these things. And if you see the DNA is one complete fragment, and this DNA is made up of 4 different nucleotides, that is adenine, guanine, cytosine, and thymine. And in different species have these 4 type of nucleotide, but their arrangement is different. In different species, different individual, we have different arrangement of these nucleotides. Due to that, we have differences. So whatever differences we are looking in our environment, whatever species, individual difference, species difference, whatever difference we are looking in the animals, all those things is just because of DNA. If you have similar DNA, then you will be similar. If you have different DNA, you will be similar. If you are related, then your DNA will be related. If you are unrelated, their DNA will not be related. So these things we can easily assess here. So once you will get the DNA with the help of different laboratory technique to extract the DNA, then we'll go for the sequencing. So before going to the sequencing, one very important component is PCR. PCR is basically polymerase chain reaction, and there is one instrument is available, that is thermal cycler. So this will basically maintain the different temperature for different time. That will help us to amplify the DNA. Because whenever we are getting DNA from any kind of samples, DNA is in very little amount. And if you use thermal cycler, if you use polymerase chain reaction, you can increase the number of that particular fragment of DNA, and that will help you for the sequencing. So you can see here in this first figure, left side, you can see this is DNA samples we are getting after DNA extraction. Then we are applying some primer. Primers are basically markers. I can say generally what we are doing, basically whenever we are using any marker or highlighter, so we are using that highlighter or marker for highlight something important in our any kind of book or copy. Whenever you are studying, we are always having one marker. So what is important for us, we are just highlighting it. Just similarly in molecular genetics, if you want to do PCR, if you want to target a specific part of the genome, we need to have primer, we need to have some marker. And if these marker will help to extract the very important portion of the genome, which is very important for you. Not all the part of genome, but some part of genome, which is very important for you, for your species identification, for looking different type of behavioral things, very many type of conservation genetics aspect. So these things are very important. So we are using primer, and those primers will help us, to amplify a specific region. So you can see here in the whatever image we have in right side. So you can see first one DNA is present, that is double helical DNA. So you can see here first it will denature. Then again you can see in third step one primer is binding both sides and they are extending. So you can see here we started with one DNA, and you can see below side after 1 or 2 cycle we are getting 4, 16, 32, this amount of DNA. So after 30 times, 40 times reaction, we are getting millions of DNA, and that will be help us to perform our experiment. So ultimate aim of polymerase chain reaction is to, if you are getting a small amount of DNA, you can target your specific DNA and you can amplify in more amount. So if you have a small amount of DNA, PCR will help you to get more amount of DNA. So this is main utilization of PCR. So still after PCR, we have only fragment of DNA. We are not able to understand how these nucleotides are arranged, because as I told earlier that if nucleotide arrangement is different, species is different. So arrangement looking is very important. How they are arranged in the DNA, that is very important. So in first step, we will amplify the DNA in enough amount. In the second step, we will go for the sequencing. So this is the sequencing step, I can say. So if you see, there are 2 types of techniques we have. One, if you left side, we are looking, that is Sanger sequencing. And another thing is next generation sequencing, that is your whole genome sequencing. This is very advanced technique now, and this is most nowadays this technique is more in use, but still people are using this targeted sequencing, that is Sanger sequencing. So as you see here in Sanger sequencing, we can do targeted sequencing. We have large genome. If you want to see, see small part of genome which is important for you, then you can use Sanger sequencing. But if you want to see whole genome of any species, how whole genome is affecting, how different part of genome is interacting with environmental change, habitat change, then you can go for the next generation sequencing. So I will little bit discuss about these things, because many of the participants, I don't think they have idea about all these things or not. So I thought if I'll give little bit more light on these aspects, so whenever new speaker will come, that will be good for them, because they will directly talk about their application of all genetics and genomics tools. So if you see Sanger sequencing, we are targeting one specific part of genome, and then we are just sequencing that thing. So in Sanger sequencing, this is basically enzymatic method for the sequencing, and this is very old technique, but this is most promising technique. Though it is taking time to get large data set, but yes, whatever sequences we are getting, that will be very promising, and we can do many more things for especially species identification. If you want to see species diversity in any national park, then you can easily see with the help of targeted sequencing. But if you want to understand how different part of genome will affect the species, how they are interacting with climate change, then we can go for the next generation sequencing, where we have we are getting all DNA, whatever DNA present inside that particular sample, and then we will go for shearing. We are shearing all the DNA, and we are putting some specific marker, specific adapter. So that adapter is just for marking that yes, this adapter is for this particular DNA. Once we will get the DNA, then we will use bioinformatics tools, and we will go for the separation of each and every DNA. So here one basic difference is there. If you are using Sanger sequencing, you can work with single sample. If you are using 2 samples in one tube, you cannot differentiate it. But if you see the next generation sequencing, here we are pulling different DNA, and after pulling, we will go for the sequencing, and at the end of sequencing, we will apply different bioinformatics tools, and we will again separate each and every DNA. So this is beauty of this next generation sequencing, where you can do lots of work within very less time. Nowadays, initially, if you go 8-10 years back, it was very challenging technology. It was very costly technology, but nowadays it is very cheap technology in India also. Many, people have such kind of facility, and they are working all these things. So in Sanger sequencing, we can use only one sample, but in next generation sequencing, we can use many samples at one time, and it will reduce our cost, it will reduce our time, and data quantity will be very large in comparison to the Sanger sequencing. Due to that, nowadays people are completely shifting from Sanger sequencing to next generation sequencing. But still, next generation sequencing have their own importance, and still this technique is existing, and that is applicable for many work in the wildlife conservation. So once you will do DNA extraction, PCR amplification, apply any one of tool of the sequencing, Sanger sequencing or next generation sequencing, then we will get sequence. So you can see here ATGC is there. So now after initially we had only fragment of DNA, we were not aware about in what fashion these nucleotides are arranged. Now we are aware about that, how these nucleotides are arranged. So if all the species will have similar kind of nucleotide, it means they are same. If they have any difference, it means they are different. So you can see here in upper row, if you see first 70 base pair, there is no difference. So first 70 base pair is same. But if you go for the second pair of 6 animals, you can see here some differences are there. So you can see here in third place, first one have T, rest one have A. So this kind of differences we will see, and we will try to understand the genetic diversity, try to understand the species differences. And this thing will also help us after applying different tools, statistical tools. We will also understand how populations are migrating, how phylogenetically they are related, and in what kind of distribution pattern they are having, and how they are interacting with the environment, and how what was their past and what will be their future. So we can see the demography of the species. So these are the basic tools, basically collecting the samples, extracting the DNA, amplifying the DNA, sequencing the DNA, and getting the information. Once you will get the sequence, then you will apply your different statistical tools. Many tools, freely tools are available in the public domain, and you can utilize it, and you can answer many questions related to the wildlife conservation. So I will discuss a little bit some important application very briefly, because our coming speakers will explain apply these tools in detail, and they will talk about many more things about different aspects of the wildlife conservation. So I just want to enlighten here how we will see the genetic diversity. Generally, maximum time we are looking the genetic diversity. What is genetic diversity? So this is very simple thing. If you get the DNA, and if all the animals will have similar type of DNA, there is no difference in the DNA, it means they have very less genetic diversity. If their DNA is different from each other, then they have some genetic diversity. So if differences will be more, diversity will be more. If differences will be less, diversity will be less. So this is very very common thing. You can see in the first sample, you can see here one sample have C, another sample have A. Rest is common. Another sample have A, sample 2 have G. So similarly, if these kinds of changes you can see in different individual, and these changes is very very common thing, because mutation is very common thing in the DNA. So if mutation will take place, there will be substitution or insertion or deletion. These are the process of mutation. So if nucleotide will substitute, then first sample is first example is very important. Suppose one nucleotide will replace another nucleotide, so that will change. And if nucleotide arrangement will change, there are possibility for change in protein, and ultimately expression of gene, and there will be difference in the behavior. If any kind of insertion or deletion will be there, some gene will deleted, some gene will be inserted, that will also change the genetic diversity of the species. So whatever genetic diversity or genetic DNA differences we have, this will arrive just because of substitution, or we can say mutation. So these are the very important phenomenon in the universe. If mutation will be there are possibility for the diversity. Apart from mutation, many other factors are involved which will leads to the genetic diversity of species. But mostly if you see, diversity is just because of mutation, substitution, or sometimes insertion or deletion. So this is the genetic diversity. If they have similar sequence, genetic diversity is very less. Animals are similar. If they have differences, so genetic diversity is there. If differences is more, genetic diversity will be more. Animal will be different, species will be different. If they have similar kind of thing, they have similarity. So similarity with DNA is always important, and based on the similarity, amount of similarity, we can identify the genetic diversity. So for assessment of genetic diversity, we need to compare the different DNA, and then we will see how they are different, and that will be their genetic diversity. So I think this is clear for you all. So another thing is, if you have many species in one area, so you can see here, if the species diversity will be there, or individual diversity will be there, genetic diversity will be there, you can see different types of animals are there. So diversity leads to their potential. If diversity will be there, their genetic potential will be there, and species will survive for the long time. So if population will mate within population, then there are less genetic diversity, and if they are breeding among the population, then there will be more diversity. This is very common thing. If you have information about genetics, so you can see here, these are the two different population of deer species, and both populations are divided by some mountain. That mountain is working as a barrier for them, but still they have some corridor that pass so that animal can move. So if you have genetic information, you can easily understand this pass is working or not. That is very important. So you can see here, one population 2 have blue color and population 1 have red color, but still you can see some red color in the population 2 and some blue color in the population 1. So this will lead to the movement. So if you are getting signature of population 2 in population 1 or population 1 in population 2, that leads to the understand the migration pattern. It means animals are migrating. Generally, we cannot see this thing, but if you are applying DNA, you can see the relationship. Then you can easily understand the movement pattern of the species. If you talk about application of these tools for different type of species, so in India, people are applied these tools for the conservation of tiger or many other species. This is one important study which was conducted in 2017. Application of genomics tools for the understanding the genetic structuring in the how many different subpopulation or population of tiger present in India. So you can see here almost 3 to 4 different populations are present in India. You can see here Central Indian population, Northeastern India, some this Western Ghat, and then this is the southern part of India. So these things generally we cannot understand. If you see all 3,000 tiger which is present in India, all have almost similar kind of morphology. You cannot at least understand how many different populations, how many different group of tiger is present. So in this study or several other studies are there, people collected biological samples, they did genetic analysis, whatever we have discussed, PCR amplification, sequencing, they applied different statistical tool, and they found that yes, how many different populations of tiger is present. So based on these things, government make a plan for the animal movement. If fragmentation is there, so government will facilitate the movement of animal, they will maintain the corridor. If population is genetic diversity is lower, then again they will do something. They will apply different resources for different population, so that will helpful for the management purpose. If you will give this information to the manager, to the forest department, to the government of India, they will easily apply their management plan, and that will be helpful for the species conservation. So this was for the understanding the subdivision in the population, genetic structuring in the population. Populations are structured or not. These things we can see with the help of genomics tools. Scientists also applied different genomics tools to understand the demography of the population. So this study was conducted for the Indian wolf, and generally, if you see the demography of any species, that is very challenging task, especially for the wild animals. How many animals are taking birth? How many animals are dying? So how they are related with each other? Understanding these things are very tough. We cannot do it in the wild condition, or if you do it for some species, that is very time-taking and tedious process. But genetics will help us to do all these things. How speciation happened, how these species diverged, how these species, develop different subspecies. These things you can easily understand with the help of genetic tools. Here you can see here how our Indian wolf diverged from the other wolf, whatever wolf population present in different part of the world, and how our Indian wolf is related with these wolf. So earlier all the wolf have similar, more common ancestor. So their ancestor are common, but just because of different changes in environment, climate change, different habitat driven changes, you can see here nowadays we have 5-6 different subspecies of wolf which is distributed in throughout the world. So earlier, if you see here, all species are were same, they were from same ancestor, but after passing time, if you go in the recent era, they started diverging, and nowadays we have different subpopulation or population or species, subspecies of wolf. So these things we can do only with the help of genetic tool. That will be very helpful for us. If you are applying traditional tool, that will be not easy task to understand these things. So genetics here help us, and I hope Dr. Yoni Aspi will discuss many more things about this wolf inbreeding and other things. Another thing is DNA barcoding. That is very important for the biodiversity assessment. That is very important to see the barcoding. And barcode is what basically whatever barcode we are looking in different grocery store, where all the stuffs, whatever materials we have, they have their own code, and based on that code they are inventorized. So similarly, we are making different DNA barcode because DNA of each and every species are different. So we are making DNA barcode, and once we will make the barcode, that will very helpful for the understanding the species distribution pattern. How many species are there? These things we can easily understand. So for that, we are taking small 500 to 600 base pair portion of DNA, and that will help give us to overall view of DNA barcoding. So you can see here, we are generally using mitochondrial DNA, which is present in mitochondria. That is COI gene. That is very universal gene. Universally, people are using throughout the globe. And this is very helpful gene to understand the genetic diversity, mitochondrial genetic diversity, and species level diversity in any population. And this is very good tool to make the DNA barcode of all these species present in the any kind of area. This is a very important thing. If you have idea about how many species are present in any area, you can assess the health of that particular population. If more species present in one area, we can assume that this area is very healthy. And if less species is present there, it means that area is not healthy. So these things you can usually do with the help of your genetic tools. After barcoding, you can do it. And these are the different steps whenever you will go for the DNA barcoding. So this is very normal. We are collecting the sample. We are going for the PCR DNA extraction, PCR amplification, DNA sequencing, and then we are getting the database of all these species. So this is very common. So these 3 steps are very common: DNA extraction, PCR amplification, and sequencing. Rest job is computer-based job. We have different kind of softwares, statistical tools, which will help us to understand these things. And all the tools are freely available in the public domain on the internet. And if you do such kind of work, DNA barcoding, so you can see here in first figure, 4 different peoples are doing morphology-based barcoding. They are identifying species based on the morphology. But you can see in the next figure, right hand side, you can see here only one person with the help of computer. They are doing tremendous job. So if you have DNA, you can do this DNA barcoding, species identification. You can develop nice database throughout the space, throughout the area, and you can within minimum time, minimum human resource, and minimum money requirement, you can easily do it, and you can get very promising data, I can say. whatever data we are getting with the help of morphological characters, DNA-based data is more promising, more important, and more what I can say, more reliable data we can say. So DNA barcoding will help us in such a way. Sometimes we are not able to collect sample of many fishes, or if any aquatic species are there, they are distributed in water and we cannot catch them. Or individually, if you are not catching them, then eDNA will help us. eDNA means environmental DNA. So generally, if you see if any fish species or any species there, if they are present inside the water, definitely they will drink water, or we can say they will be in contact of water. If they will be in the contact of water, there definitely there will be some part or some part of body or anyhow some biological material will come out and that will flow in the water. So if you collect the water sample, of any river or any pond, then you can easily understand the species diversity. How many type of species are present? How many fishes are present? How many amphibians are present? All those things we can easily do it. And you can see here nowadays eDNA barcoding, meta barcoding we have, where we can use pool of DNA. If many DNA is present in one pool, we can do meta barcoding, and then we can differentiate it with the help of different type of bioinformatics tools. So you can see identifying the biodiversity of any pond or any kind of river system is very tough task. You need to collect each and every species, then you can say how many species are present. But if you are applying eDNA technology, you just collect a water sample from different part of that particular pond or river, go for the eDNA meta barcoding or eDNA barcoding. Different tools are available. You can easily assess the diversity of that particular ecosystem, or pond ecosystem or river ecosystem. So that is very helpful, very cheap, very convincing, and most reliable technique where we can easily understand the diversity of species inside the habitat. That is only possible with the help of molecular tools, especially genomics tools, especially eDNA barcoding. Sometimes if you have if you have soil and many kind of species are present in the soil, if you are getting sediment in the any pond, there are also many different species are present. So you can easily collect the sample, go for eDNA barcoding, go for the meta barcoding. You can easily assess the species diversity there. If you want to understand the diet of any species, that is very important, because understanding diet of any wild animal is really tough. What they are eating, we are unable to know. People have some morphology-based technology. They are using microscopic-based technology, so they are making slide. And if any undigested remains is present in the fecal sample, then we can see all those things, make slide with the help of hair pattern or some, if you are looking at the plant, then dicot, monocot. So we can see little bit, 10% we can see what kind of species they have taken. But 90% species we cannot see with the help of normal morphological tool. So in that, in this condition, or what you can say, this barcoding will help you. You just take the fecal sample, extract DNA of all the fecal sample, all the DNA, go for the meta barcoding, and after doing meta barcoding, separate it. You will get almost 90% information about diet. So if you are using morphological method, you are getting only 10% information. If you are using meta barcoding method, you will get only almost 90% information. That will be more promising, more convincing, and that will help us to make some plan. What kind of things, what kind of material those animal is eating, we can provide them those things, or we can anyhow help them to get all those kinds of material. So that is very helpful for the wildlife conservation. If you are applying these tools, apart, traditionally we have many tools, but those tools are very time consuming, money consuming. They need some resources, especially human resources. And if you are applying this meta barcoding tools, they are need less human resource, and they are giving very promising data. If you want to see the gut microbiota of any species, you must have heard about gut feeling. So gut feeling is very common. Whatever your gut is feeling, it means same thing you are also feeling. So if you want to understand what kind of microorganism present in our gut, you can easily do it with the help of meta barcoding. Just collect the fecal sample, the compulsion is fecal sample should be very fresh. If you are collecting very fresh fecal sample, you can easily understand the importance of this technique. You can easily understand how many type of microflora is present in your gut. And if you see the spectrum of microflora, it will differ habitat to habitat. If one species will live in different habitat, their gut microbiota will be different, because they are living in different kind of environment. Their food is little bit different, and if environment is different, food is different, definitely gut microbiota will be also different. So these things we can do only with the help of metabarcoding. This genomics tools is very important. Otherwise, we cannot see the health of. So with the help of these tools, we can also see the health of species. If microflora is enough, different diverse microflora they have, it means they are good in condition, their health is good. If their microflora diversity is very less, it means that animal is not in healthy condition. This is very common thing. This is well-known fact for human also, but people are now applying for the different kind of animals, and we are getting promising result. If you are getting archaeological sample, because if you see the historical sample, if you are getting very old sample, then you can do this metagenomics tools, and you can easily see how many species are there, what was the possible reason for the extinction, how they started decline, how they diverged. All those things you can easily understand. So with these things, I would like to stop my presentation, and I hope all the audience now will have some insight about use of DNA, how you can use the DNA, different tools, what kind of tools are available in the public domain which we can apply to wildlife conservation, and how especially what we can say, these techniques are less time consuming. Nowadays, these techniques are very advanced. They also involve very less amount of money, and whatever information they are generating, that is enormous. So if you compare traditional method and their investment, if you see the modern method, modern genomics method and their investment, so genomics method is more promising, more convincing, and more reliable things. So with these things, I would like to stop my words here, and thank you all for listening carefully. And if anyone, any query if you'll have, you can ask. I would be happy to answer you. Any query?
Thank you, Dr. Sujit. Wonderful presentation. So participants, if you have any query, you can type your question in the chat box, so we can forward to Dr. Sujit.
Okay, so sir, I think Dr. Yoni joined or not?
Yeah, Dr. Johnny joined.
Okay, my things is little bit okay. Stop sharing then. Okay, that's fine. So any participant, if you have any question. Oh, Yoni. Hello, Yoni.
Hello.
Good morning.
Nice to see you all. Good morning.
Good morning. I think almost 10 a.m. in the Finland. So
Yep.
I think yes. And nice to see you after a long time.
Likewise.
Yeah. So our next speaker will be Dr. Joni Aspi. Generally in India, we are calling Johnny Aspi, but in Finnish, J can be called as Y. So he's Joni Aspi. And Joni Aspi is currently working as a Director of Ecology and Genetics Research Unit of Department of Biology, University of Oulu, Finland. And I would like to say here that he was my PhD supervisor. So I spent lots of time with him, and I learned many more things. Whatever I was discussing in my talk, many more things I have learned with Dr. Joni. And I am very thankful to Dr. Joni also because he accepted our invitation and he's spending time for his presentation. So Dr. Joni is currently working as a director, and he's a professor in the department. He's one of the senior most faculty in the department. Apart from department, he's also looking Zoological Museum and Botanical Garden. And apart from his research and other thing, he's enjoying cycling and mountain climbing, especially skiing in the snow. It's because in Finland, snow is very common thing. So Joni is very, he's always spending at least his weekends for skiing and cycling and all these things. So, Dr. Yoni, I think I would like to invite you for your talk. So please.
Yeah, thank you very much for the kind invitation to the event. And I am going to talk you about, so can you see my presentation now?
Yeah. Now it is visible.
Yeah, sorry about, is it then in presentation mode or?
Just 1 minute. Yeah, it is in presentation mode, I think.
Okay, so yeah, I'm going to talk about inbreeding in Fennoscandian wolves in Scandinavian and Finnish wolves, a little bit about their history and level of inbreeding. And both Finnish and Swedish population has gone through quite narrow bottlenecks. This is showing the number of wolves killed in Finland during the last 150 years. It has estimated that about 23,000 wolves has been killed in Finland during that time. So in the middle of the 19th century, the species was quite common in whole country, and it has been estimated that there has been over 1,000 wolves in Finland. But in late 19th century, organized riots started to remove the species from Finland. And you can see from this graph that during in 5 or 10 year periods, several thousand wolves were killed in Finland. And in 1920s and 1916s, 17s, there were only a few wolves in Finland. But situation in Sweden was even worse. Their wolf died altogether in somewhere in 1960s. So there were no wolves in Scandinavia or Sweden. And actually after 1990s, the wolf was started to increase. It was not allowed to kill wolves anymore, except in the reindeer herding area in northern part of Finland. And the population started to increase. This is showing the minimum population size. And it increased until 2006 from 5 packs to 25 packs. After that, hunting of wolves were allowed in several years, and then it started to decrease. And but after that, it was again protected. No hunting allowed, and population started to increase. And nowadays we have about 300 wolves in Finland. So the wolf disappeared from Sweden in 1960s, but then in 1983, one pack appeared here in this part of Sweden. And this pair probably came from Finnish or Russian population. At that time, the stronghold of the Finnish population was here in eastern Finland. Nowadays, the stronghold is actually here. The population has increased and spread to west. Okay, so from 1983 to 1991, there was only one family group. And in 1991, a male appeared to the Swedish wolf population. It was also coming from from this Russian Finnish wolf population. And it was very successful, and the population started to grow exponentially. And then 17 years later, then there were 2 new immigrants, which appeared to 2006, 2007, and they were breeding from 2008 to 2010, and it again increased the growth of the population. And 2012 there were already over 300 wolves, and nowadays there are about, though there were actually 400 wolves. And this is also showing the inbreeding level of the individuals during their first breeding. But let's look closer, to the pedigree of the Swedish wolves, because the population is in rather restricted area, and they have, in Sweden, they have a good collection of samples, cats and killed wolves and so on. So they have been able to build the pedigree of the Swedish wolves. This is showing the first bear in 1983, and then the male from 1991, and then here again the 2006, 2007 individuals. So there are altogether 5 individuals coming from Finnish or Russian population. And this is showing, they were assumed that there is no inbreeding in those individuals. But on the basis of pedigree, this first male actually paired with his daughters, and they produced some offspring until this second male appeared in the population. But these blue ones are only lightly inbred individuals, but these red ones, their inbreeding level is already something like over 0.4, and the gray ones 0.5. And okay, in this shows the development of the inbreeding level. The inbreeding level reached 0.25. The mean inbreeding level, which is similar than the pups of full sisters or father or parent and offspring. But it increased over that to 0.3 here, and then when the 2 new individuals appeared in the population, it started to decrease and decreased from 0.3 to 0.24 in 5 years. And this is showing all the immigrants which has came to Sweden until 2015, and you can see the first pair here, and 1991 pair, and 2006 and 7 individuals with green. But there are a lot of red dots here. There are individuals which were not breeding in the population. And 2014-15 there was one pair here, which was translocated with the airplane to the breeding area of the Swedish wolf. Here in the northern part of the Finland, there is not wolves, because this is reindeer herding area, and the wolf is not protected here. And the stronghold is nowadays here in the southeastern Finland, and the Finnish wolf population distribution area is here in northern Sweden. So it is very rare when wolves from this area are able to come here. So how about then Finnish population. Genetic diversity in individual level we have is shown in this picture and year of the birth of an individual here in different years. And you can see that this multi-level heterozygosity has been decreasing despite the fact that the population has increased. This is probably because the Russian wolf population size has been decreasing and there is less migration from there. And it has continued this decrease of the heterozygosity, even though it is still quite high. But it doesn't actually tell us anything about the actual level of the inbreeding among wolf individuals. And we had tried to construct the pedigree of the Finnish wolf population. Yeah, it seems to be possible in local level. So when the wolves spread to the southwestern Finland, there were only limited amount of individuals in this area here, and it was possible to build a pedigree on the basis of scats collected by local people. But if we are trying to do it in country level, our results look like this. So there is this main pedigree, and on the basis of this we can estimate the inbreeding level of different individuals in this pedigree. But there are several families which are not connected to this main pedigree. So it is easy in Sweden when the population distribution is quite restricted and there's not very much migration outside of the population, but we are getting each year a few immigrants from the Russian population, and then we have got a lot of missing data in our sample collection. It is not so effective than in Sweden. So on the basis of pedigrees, we actually cannot estimate the inbreeding levels of the individuals. But as Josette has shown us, on the basis of whole genome sequencing, it is when it has come cheaper and cheaper, it is possible to estimate the inbreeding level on the basis of runs of homozygosity or rows, which are long stretches of homozygous genome. And this is showing an example of some chromosomes and individual which seems to have that kind of row in chromosome 4 here, and it is in the chromosome 4 there seems to be about 8 centimorgans of those long stretches of homozygous genome, and altogether in this individual there were 46.7 megabytes of those rows, and if you are comparing it to total genome, it seems that the detected rows account about 1.7% of the reported genome. And this is actually part of my genome. So I'm a inbred at that level, but it is quite typical to Western population, this kind of figure. But so on the basis of these rows, we can estimate inbreeding level of an individual, and it seems to be correlated very well, the inbreeding level measured on the basis of pedigrees, and it is also correlated with multilocus heterozygosity. And we have been actually trying to estimate it also in the Finnish population. Jenny Harmoinen, who is our PhD student, has been first tried to estimate it using an Illumina's Canine bead chip, which are commercial chips, and where there are more than 170,000 evenly spaced and validated SNPs. And on the basis of this analysis, she was able to estimate and inbreeding levels of some individuals of the Finnish wolf population. And here we have some examples of the inbreeding levels of some individuals. One of these cases is this pup of Ugri and Maija. Ugri and Maija is a father-daughter pair, and we expect that the inbreeding level in that kind of pairing would be something like 0.25, and in this case it seems to be quite close of it. But we find also several other individuals with high level of inbreeding, and especially this one in the upper row of the table, it has level 0.38. It is very high, and actually this is probably an immigrant from Russian inbred population. We are also getting some immigrants from Scandinavian inbred population, but very rarely. But it is more this Illumina chip is a good tool, but maybe it's financially more productive to make whole genome sequencing using Illumina platform. And for example, Hans Ellegren from Uppsala has done it for 87 individuals in the Swedish wolf population, and this is showing the results of the in the lower part there are some quite inbred individuals, and this the upper part, so the inbreeding level, this FROF in the immigrant individuals, which were in their pedigree assumed that they are not inbred at all. But it appeared that actually some of the individuals were moderately inbred. Remember that all of these individuals were not actually breeding. But there were, and this figure is showing the 83 chromosomes of the wolf and the heterozygosity level in each chromosome. And you can see that there are individuals where there is not whole chromosomes where there is not at all any heterozygous. They are all homozygous. And the mean FROF among those 97 individuals was 0.27. It was a little bit higher than estimated on the basis of pedigrees because of this strong assumption that the founder individuals were not inbred. So we teamed with Hans Ellegren and sent him about 100 samples from the shot individuals from the Finnish population. Quite many of them were here from the reindeer herding area, where you can freely shot the wolves who immigrate there. And this is showing the 10 most inbred and 10 less inbred individuals in the Finnish wolf population. And we can see this individual in this lower row was that Vasily, who is the pup of Maya and Ukri, who were father and daughter, and the estimated level of inbreeding is 0.244, which is very close to expected 0.25. And you can see that in that individual, there were some very, very long regions without not any heterozygosity. But there were, in addition to that individual, there were other inbred individuals also in the Finnish population. And this is here, this figure here is showing the mean inbreeding level, which is 0.45, which is actually not very high if you're comparing it to the inbred Swedish population. And if we are looking the breeding status of the individuals, this O here means that the individuals were not breeding, and 1 means that the individuals were breeding. And you can see that the breeding individuals has much lower inbreeding level compared about half of that what is for the inbreeding level from the non-breeding individuals, and the difference is significant between non-breeding and breeding individuals. But what is more worrying in this Finnish population, that if we are looking the inbreeding level of the individuals and then birth year, it seems that the inbreeding level is increasing. It has been increasing from middle of 1990s to 2016. This case here is the known Vasily, the known father-daughter pup. But there are others with high inbreeding level, which seems to be concentrated here in the later years. And this regression seems to be significant. So it's a bit worrying that even though in the Finnish population, the mean level of inbreeding is not so bad, but it seems to be increasing. And what's up? Why are we so worried about this inbreeding? As Dr. Singh's already told us, this inbreeding can cause inbreeding depression. And it has been shown, for example, in this highly inbred Swedish population. This figure here is showing the inbreeding coefficients of the pups, and then pup mortality, which is measured as how many, what was the number of pups in the pack in the winter. Here, when the inbreeding level is 0, the number of pups was 6, and here, when the inbreeding level is 0.25, which is offspring inbreeding level of parent offspring or full ships pups, and it seems to be something like 3. And here, when it is 0.4, it is maybe 0.4, it is maybe only 2 individuals. So it seems that it is decreasing the pup's survival. And then Jannike Räikkönen has studied congenital anomalies in small isolated populations in this Scandinavian wolf population, and also in Isle Royale population in US. It's an island population where there were which was very small and in breadth. But Jannike, who presented has her doctoral dissertation in 2017 here in our university, in our research group. And she was studying the Scandinavian wolf populations and looked for this kind of anomalies, like this is showing normal canine occlusion in the upper figure compared to the malocclusion in this individual, and in the right figure there's a pronounced overshot and uncomfortable jaw closure in this individual. She found quite a lot of these congenital anomalies in the Swedish wolf population, but actually she didn't find any from the Finnish population. So it seems that we don't have those anomalies yet in the Finnish wolf population, even though there are quite a lot in the Scandinavian wolf population. And what she also showed on this publication and in her thesis, that this is showing the year of the birth of an individual and then incidence of these anomalies, and it seems that the incidence of these anomalies rose from 13% to 40% during her 23-year study period. And many of these anomalies are very probably associated with inbreeding, some form of genetic deterioration. So actually, Jannike is showing that even though genetics is very important nowadays for the management of the populations, but the morphology still could have some and very good tool for studying effects of inbreeding using classical museum collections. And something about the recent development, actually last year in May 2022, the Swedish Parliament announced a goal to reduce the Swedish wolf population from about this 4,000 to about 200 individuals. And then we actually write together with Linda Leigre and Fred Allendorf and me and other wolf researcher letter to Science, where we claimed that this planet cull endangers the Swedish wolf population. So that we told that the wolf numbers are not large enough to sustain healthy population, and this culling is not supposed healthy for the wolf population. Anyway, the Stockholm government authorized shooting of 75 wolves, and in this year hunters have shot 54 animals, which is the largest ever cull in the Swedish population. So instead of 200 individuals, only 54 individuals were shot, but maybe that is not anyway good for the wolf management in Sweden. But yes, so there has been quite many people involved in these studies. These people are from our research group. Eva Jansson Eva is actually nowadays in Trondheim in Marine Research Institute, and Laura Quist is another senior scientist in our research group, and Jenny is our PhD student, and Suvi Ponnakas is a postdoctoral student who is now looking closer to the whole genome genomes of our wolves, looking what kind of mutations there are and what kind of deleterious alleles there are. And Ilpo Kojola is the Finnish Natural Resources Institute and has been a long time in our close cooperation in wolf research. Han Ellegren, of course, is a grand old man in wolf studies from Uppsala, and Mikael Åkesson from Swedish Agricultural University Grimsö has also been working with wolf inbreeding in Scandinavia. And so, do you have any questions now?
Participants, any question? If you have, you can type here. I can ask Dr. Yoni. So by the way, Dr. Yoni, I have some question. Generally,
Yes.
if you are looking, inbreeding leads to the inbreeding depression.
Yes.
But is it individually specific or population specific?
Yeah. Actually, when Jannike was studying those congenital anomalies, she didn't know what was the individual level of inbreeding of the studied animal, because for some reason the other research groups were not willing to tell it to her, even though skulls are nowadays in the Swedish Natural History Museum, where Jannike is working nowadays. But probably it is also in the individual level.
Okay. Okay, one more question. This is a little bit different, but it is in perspective of India. Now you must have heard about tiger population in India is increasing.
Yeah.
We have almost more than 3,000 tigers.
Yes,
but
indeed.
now some conservationists, they are raising their voice for the culling of tiger because lots of conflict going on. So what is your view about all these things? Because you have written this, you people oppose the slaughtering of these, especially wolf.
Yeah, in Sweden, and there are also some plans to start reducing the Finnish wolf population, which is nowadays about 300 individuals. But as if you are looking this figure, which is showing that inbreeding level has been increasing in the Finnish wolf population, it is not very healthy to start culling them. And I don't know what has been the development of the inbreeding level in the Indian tiger population. Maybe if you can study that, it could help to show that.
So before doing these things, we need to understand these aspects. Then we can see.
Indeed.
Yeah, okay.
So are there any questions in the chat?
No, sir. I think there is no question. So,
okay.
So thank you very much, Dr. Yoni, for giving your nice talk. I was aware about many more things because I'm already, looking at all the updates. But I think for the participant, that was very informative, how we can apply genetic tools, because many participants they are basically from environmental science background. So they are not aware about the potential of genetic tools to understanding such kind of conservation aspect. So I hope your talk must have enlightened something, and that must be helpful for the all the participants. So thank you for your presentation, and I'm heartily thankful to you because you accepted our invitation to present here, and hope for future cooperation and all these things. And one thing was there, as I discussed earlier, that one of the students was applying for this 14 Future World program. So now she got selected, sir.
Okay,
so,
that's
so
nice.
her proposal with Stephen and Marco, it was accepted, and
that's great.
She is planning to go there in September. So she is planning for her, residential permits and all. So hopefully we can,
Okay.
continue our cooperation with the help through Shreya and other students.
Okay. Okay. Thank you again for inviting me here. It was my pleasure. So
Thank you, sir. Thank you very much, sir.
Dr.
Thank you.
Morning. Nice to see you after a long time.
twice.
Dr. Randeep is, I think, yeah.
Yes, after 2007.
Yeah.
So
Okay, sir. Thank you for your talk. And now we will move for the next talk. So I think Dr. Pandey is our next speaker. Dr. Pandey had joined or not?
Dr. Punit Pandey joined.
He's
Yes,
so yes, now. Hello, Dr. Pandey. Good afternoon. I think this is you are unmuted. So I think
Is audible. Good
yeah,
evening.
now it is audible. So I think good evening to you because this evening time in Korea. So so
it is a noon time.
so I think we can start your talk. So for the participant, I would like to give little bit introduction of Dr. Punit Pandey. So Dr. Pandey is working in the field of wildlife conservation since last 13-14 years, and he worked here in India for tiger conservation. Especially he did his PhD on Ranthambore tiger, and he did PhD from Wildlife Institute of India. Nowadays, he is serving as a senior research scientist there in Seoul National University, and he's continuing his work on Russian this leopard and Amur tiger, this Chinese tiger. So he's continuing his work, and especially he's applying recent genomics and genetics tools for the conservation. So I would like to invite Dr. Pandey to give his talk. Dr. Pandey, you can start your talk.
Thanks, Dr. Sujit and Dr. Randeep for inviting me, and good evening to all of you. So for the benefit of the audience, I made this presentation of mine a little more simple, where I would like to share something related to how technical and scientific cooperation can be done in the field of wildlife sciences. And my focus when I'm talking about the technical and scientific cooperation, I'll be focusing on the genetic tools and techniques. So when we talk about the technical and scientific cooperation, there are two major requirements. One, partners should have certain requirements in terms of the knowledge, expertise, or funds, resource, etc. And the another partner should have the enough resources to provide those kind of a support. So technical and scientific cooperation can be interdisciplinarily. For an example, a person from a genetic background can have a cooperation or a collaboration with an ecologist or a policymaker to find out the ultimate answer, or it can be an inter-institutional, wherein two different institutes working in these specific domains are joining hands together for the benefit for the biodiversity conservation. Or it can be an intergovernmental, wherein two different countries, government bring together the scientists and the experts on one particular platform. Specifically, this kind of a cooperation is more important for a species that have a long range and where their distribution is overlapped over the different countries. So I'll be trying to explain you with the examples and everything in subsequent slides about how this kind of a cooperation is achieved and how I am working in all those aspects. So as Dr. Sujit have mentioned, I did my PhD on tiger genetics from Wildlife Institute of India. Currently, I am working as a senior scientist with Conservation Genome Resource Bank for Korean Wildlife at Seoul National University. Also, I am associated with the National Institute of Ecology, National Institute of Biological Resources of the South Korean Ministry of Environment. I am also serving as a research advisor with Bangladesh Forest Department for their Sundarbans Tiger Project. And I'm also serving as a vice president to the Protect India Foundation. This is an NGO aimed to promote scientific science-based conservation methods and education in India. During the course of there are some. So I am here since 2020. And over the past 3 years, I have established the research stations in 3 countries, wherein my teams are working there in collaboration or in continuous support from South Korea. One station is based in the Land of the Leopard National Park in Russia. Second one is in Sundarban landscape in Bangladesh. The research station is basically based in the Dhaka in the Wildlife Crime Control Unit. And third one is in the Jilin province of China. So Jilin province of China and the Russia are very near. And this is the one area where you have the Amur leopard population. Amur leopard is one of the highly endangered species, critically endangered species, with less than 100 individuals surviving. And the landscape which these 100 individual covers is more than 4,000 square kilometers. So such a long forested landscape. So we need to have a cooperation among the different countries. Unfortunately, because of some administrative issues, the one of the range country, that's North Korea, is not included till now in the project. But we hope with the collaboration with the China and the Russia, this species can be restored and fluctuate. So nothing to tell you about this. This is already covered. We are living in the 6th mass extinction phase. This mass extinction event is driven by the human activities. That's why it's called the Anthropocene. And this is like a exacerbated rate of the species extinction. So when we talk about the species extinction, one should keep in mind species always extinct. It's not a first time. As species evolve to new species, certain species bound to extinct. However, when the rate of extinction becomes multifold increase compared to the background rate of extinction, that is like a stable extinction rate, then we call it as a mass extinction event. So this is a slide from the, I've taken the data from the IUCN report, which clearly suggests that the past few centuries, the rate of extinction has been exponentially high across different taxa. So if you see here, like other vertebrates like invertebrates and things, you find less extinction rate, but that's not an exact situation. Because of data deficiency, many of these species we are not sure or not know. As I already mentioned, like we are living in the Anthropocene, so it's all about the human population expansion, change in the habitats, fragmentation of habitat. All this is causing the increase in the extinction of the wild species. So once a species extinct from the wild, it creates a lot of problems across the ecosystem. Ecosystem balance is disrupted, which in turn increase the rate of extinction of many other species. So currently for a wildlife, we have a lot of challenges, which I have pointed out in major 3 categories. One is like a habitat alteration, like habitat loss, fragmentation. Second is like population status. Many species, we are not sure about their population. Before making any conservation policy, one should know the population status. How much is the population? How much is the carrying capacity? And how was the population in the past? And third one is the conflict and poaching. Like in case of conflict, when humans lives very close to the forest, or when the humans encroach the forest, there are the incidents where their interaction becomes negative, wherein animals are killed, persecuted, and sometimes even after killing, they are traded. So in this slide, you can clearly understand what happens when a population decreases to a small and it becomes fragmented due to the number of reasons ranging from habitat loss, overexploitation in terms of hunting, poaching, exotic species arrival in the ecosystem, and pollution. So once the population becomes small, they are trying to breed them among their relatives. That increases the inbreeding or leading to the genetic diversity loss. Genetic diversity is directly linked to the individuals' potential to survive in the harsh climatic conditions. So once genetic diversity is lost, they have a reduced adaptability or a survival and even the reproduction. This in turn reduce their number and finally leads to the extinction in many cases. As Dr. Sujit and Dr. Johnny already mentioned about it, there are number of applications of the genetics in wildlife conservation and management, number like species biology, taxonomy, forensic investigation, breeding programs, species recovery, etc. So DNA tools can be used in multiple ways to provide the effective information that is required by the wildlife managers for the management of species. In this briefly, I've just tried to explain why DNA is important. So DNA codes the information that translates into the protein. And with this protein, what a species look, what the diversity they have, what the characters they have, morphological, everything is governed by the proteins which are expressed. So any change in the DNA leads to the change in the RNA and then the protein, or any reduction in the diversity of the DNA will ultimately leads to the reduction in the expression, that in turns reduction in the expression of the different traits within a population. So we work on different kind of the sampling methods when we talk about the wildlife, because it's very difficult to capture live animals. So many times we work on non-invasive, non-destructive samplings. Most of my work is related to non-invasive, non-destructive sampling. So this is a proper definition of conservation genetics. That's the application of genetics to preserve species dynamic entities capable of coping with the environmental changes. One should keep in mind, when we talk about the genetic management, we are not talking about increasing the number. Our ultimate goal is to increase the overall genetic diversity. So if you have a related individuals, their number is increasing, that population can collapse any point of the time. So as to avoid those kind of an events, one should always look into the viable options, viable potential individuals to be bred either in captivity or within a population. Suppose for an example, if a population is small, isolated, in that case, efforts are made either to resupplement the population via reintroductions or facilitate the animal movement or exchange through the establishment of corridors within the populations. So basically two different classes of the molecular markers that are used. And PCR, Dr. Sujit has already explained, this is the one of the basic techniques that we use after DNA extraction. It amplifies the targeted region to the multifolds, and that is able, with this multifold increase amplified DNA, we are able to check the DNA variation, either using the electrophoresis based on the pattern of the bands or through the sequencing method, where we actually identify the sequencing pattern like the pattern of the nucleotides within the DNA, or using the microsatellite markers. Microsatellite markers are the repetitive regions within a DNA, and these are the most potential markers that are used for the population genetic studies, wherein our interest is to know who is the relative, how the gene flow is happening within the population or among the population, how the animals are moving from one place to another. For all those issues where a recent information is required about a species, microsatellite markers are much more informative compared to the mitochondrial markers. So now coming to some examples through which I'll try to explain how these markers and techniques can be used. One first example is about the leopards of Korea. So like India, Korea once had both tiger and leopard. Tiger went extinct in 1930s and leopard was last seen in wild around 1970s. Since then, IUCN has also declared leopard extinct in South Korea. Currently, a small leopard population exists. That's the only leopard population that is in the Russia Far East in this region. This is also one of my research area or a research station. And this leopard population is now expanding. So we are trying to look how possibly the North Korean Peninsula also can be repopulated with the leopards in future. But before starting about these kind of a project, one should first confirm or identify what was the phylogenetic affinity of the leopards that were once lived in the South Korea. One of the challenges that you find while you were working in a country like South Korea, which had a long history of occupation and wars, you hardly find any reference sample in any museum within the Korea. Similar was the case with this leopard also. There was no sample available, so no study was ever conducted on the genetics. Fortunately, we found one hunter who is having a possession of leopard skin rug. That this animal was killed in 1935 in a place called Jirisan. This Jirisan National Park is now well known for the bear population. The bear restoration is already finished in this region, and this population is flourishing. Historically, there were leopards there, but now there is no leopard. So we undertook this kind of an investigation to identify what is the phylogenetic affinity of this leopard, and then suggest what might be the potential candidates for the reintroduction of leopards in South Korea, as South Korean government is keenly looking for a leopard restoration in South Korea. Very simple phylogenetic study was conducted. We amplified the mitochondrial DNA, and we downloaded many sequences of different leopard subspecies from the NCBI databank online. And then we constructed the sequence. And what we found it out is like this ORI1, ORI2. So this is like a Amur leopard. So this Korean leopard was also an Amur leopard. Although it was obvious, but we need some kind of a scientific document to prove it. With this proof, it was established that the animal that went extinct in South Korea, the leopards, were actually the Amur leopards. We published this study in PRJ, and we submitted one report for the recommendation of reintroducing the Amur leopards. So Amur leopard species program, conservation breeding program is there. That's called EEP program in Europe. So they have some animals, they are breeding there. So we till now made an agreement with the European counterparts. They are ready to provide us like around 4 individuals, but still we need to find a proper place for their breeding and the management plan, how these animals will be restored in the wild. So still a long way to go, but background is established so that once in future this species can be restored back in South Korea. Another story is about the black bear. So as I mentioned, like almost entirely all the large carnivores or the large predators are now extinct from South Korea. So similar was the case of the black bear. So black bear went extinct in 2000. The last time it was seen in the 2000. Since then, it was established again, the population was established in the Jirisan National Park. So they say like they had like 5 individuals surviving in that region, but still that was the question in mind, like whether they were the native Korean black bears or not. Because in Korea, they had, after Korean War, like say 1950 onwards, because of the economic situation, they started the animal farming. So they imported a lot of animals, bears from the Russia and China, and start farming. Many times these animals escaped from these farms and went into the wild. So we were not sure like these animals were basically from South Korea or they are basically some animals who strayed out from farms. So this was very ambiguous. And moreover, there was no, again, the similar case, no sample was available. So fortunately in 2016, some ecologists were doing some survey and they find it out one in limestone cave, some bones which were suspected to be of a bear. And we undertook this study. Our main question was, are these bones belong to the South Korean native bear? So I'll try to explain you how we'll come to the conclusion that these are the bones of the native South Korean bear or not. And If yes, which is the most recent common ancestor to them? To answer this question, one thing is like, if I want to know what is the phylogenetic ancestry, the study is very similar like that of a leopard. Like you amplify a DNA and then try to analyze the DNA sequences with the downloaded sequence from the NCBI, make a phylogenetic tree and try to see the molecular affinity, which subspecies it is more related, and then you identify. But as I mentioned, from 1950 onwards, there is a inflow of bears for farming. So it becomes like very difficult to say like whether these bones were actually animal who was farmed or not. So we made our hypothesis like, let's calculate the age of this using the radiocarbon dating method. And if this bones belongs to an animal that has existed before the Korean War time, that is before 1950, then it is like almost sure that these are the native animals, because before that there was no any evidence was there that animals were introduced back in South Korea. So like I told you, like we amplified the cytochrome B gene, COI gene. First, we identified the species. We identified like it's a black bear. And then we did the molecular phylogeny using the D-loops region, and we identified that it belongs to the Asiatic black bear group, which is very similar to the animals that are currently surviving in the Russia, China, and North Korea. So our findings suggest that, yeah, the group is same. Korean Peninsula has a historical connectivity to the Russia, Far East, and Northeast China. And probably these all the region had a one contiguous population in the past. Second thing we did like radiocarbon dating, and based on our results, we identified that animal must have lived before 1950s. And with this, we confirmed that this is a Korean leopard. So this study was very much important in terms of the management of a species, because in South Korea, wildlife is subjected to two different ministries. One is Ministry of Environment and another is Ministry of Cultural Affairs. Because like they make species like endangered. So the word endangered is associated with Ministry of Environment, and they have a typical term in Korean, which is like a species of a national monument status. So like otter, bear, so all these species are also something. Both these ministries mention the leopard with two different names. One says Asiatic black bear, another had another scientific name like Ursus thebenthus is the black bear, and the Ursus voltioni, that is another scientific name which is used by the Ministry of Culture. And they always insist that these black bears of South Korea were different, and they are always being vocal about the issue, like they don't want reintroductions or the resupplementation of the population from the other countries. So with us, with this study, we try to bridge the gap between the ministries and solve their issues. And this study is a very good example of a technical and scientific cooperation among different institutions, because this study had a participants from the 3 different institutes having 3 different expertise. So one institute was involved in the ecological survey, another was involved in the radiocarbon dating, and another institute was involved in the genetic analysis. And none of these institutions were having the independent expertise altogether. We published this article in Animal Cells and Systems. Another study I would like to share is about the water deer. So water deer is something like a chital deer that you find it in India, axis, but it has, the male has the teeth, protruding teeth, which is more similar like a musk deer. If on the left hand side, you see the historical range and the current distribution range of the water deer. So if you see there, here they mentioned, like the IUCN mentioned the distribution of water deer, but there is no official record available from the North Korea for a very long time about the presence of water deer. So people are not sure about it. And they talk about the two different water deer subspecies, Chinese water deer and the Korean water deer. Out of their historical or the former distribution range, in past few years, water deer was spotted in the parts of Russia, Far East and Northeast China. In these regions, they have a lot of musk deer and hunting is permitted. So sometimes hunter hunt deer and they mention like a strange musk deer because morphologically that looks very different from a musk deer. So for like few years, this debate goes on. There is some special type of a musk deer is found in this region. But when the investigation was undertaken by some morphological experts, they suspected this might be a water deer. And later it was established that it was the hunted animals were water deer, and the investigation was published in Russian some journal. So in this study, we conducted the molecular phylogeny of the water deer and tried to find it out first what these animals are, and then if they are water deer, what is the possible way of their dispersal or how their range has been expanded. Because in much of the Northeast China, the information about wildlife is very vague, very less is available. So we did a molecular phylogeny in a similar way as I mentioned previously, and we confirmed the samples that were collected from Russia. This Russia Far East, this is Jilin is China. So all these animals are water deer. The second question comes, are these water deer belongs to the Chinese subspecies or a Korean subspecies? So in Korea, we have a lot of water deer. So sampling was not an issue. A lot of research has already been undertaken in water deer about water deer in South Korea. In fact, water deer is like a vermin species you'll find everywhere. So we investigated, but unfortunately from the China, for Chinese water deer, we had only 2 sequences available. And then we did the molecular phylogeny, and our results were very inconclusive. We were not able to separate the water deer, Chinese water deer and the Korean water deer into different groups. This was the condition before also. Some researchers have published this study. They were also unable to differentiate between the Chinese and the Korean water deer subspecies that were defined morphologically. So the question arises, is the subspecies status requires revision? Is they are true subspecies, or these subspecies definition needs to be revisited, or this kind of information or the results that we are getting is based on because of the data deficiency, as we had a very less information about the Chinese subspecies, this region. Only 2 sequences are available on NCBI. So now we are trying to contact to the, because from China, it's very difficult to get the sample from this region. So we are trying to contact the United Kingdom authorities because they have a lot of water deer. These water deers were reintroduced from this part of China, south part of China. And so these are like the Chinese water deer. So once we get these samples, then we'll be able to understand what is the main cause. Third, I'd like to share about my current project. It's about, the title of the project is about Asian big cat conservation through technology sharing and the use of unified DNA typing methodology. This is the CBD funded project and also partially supported by the Ministry of Environment, South Korea. This is the only project in Asia about the conservation of big cats, wherein we are talking about the training, joint research, resource sharing. And this project has been now a model for the technical and scientific cooperation among the different parties. So one may ask me a question, why big cats? So big cats, like before that, I would like to say we are making this kind of an infonet, ABC infonet, like Asian Big Cats Network. So we are in touch with like more than 10 countries, and we are keep on trying to expand our group. So few months back, even our Honorable Prime Minister Modi talked about Asian Big Cat Alliance. So this is a similar kind of an Big Cat Alliance, but only majorly focusing only on the Asian Big Cat range countries because of our limited resources that we have. So one can ask, why big cats? Why we are only focusing on big cat? There are several other endangered species that needs protection. So big cats qualifies like few points by which you can say that if you protect them, the entire ecosystems can be protected. First, they are the hypercarnivorous apex predators. So many species distribution is indirectly or directly governed or influenced by the presence of big cats. Their population is declining across the areas. Wherever their population is increasing, that increase is from the historical steep decline. Like for an example, now the population of tigers are increasing, but if you look into like say about a century back, what was the tiger population in India? Still the population is not more than 5%. It's less than 4% at the moment. So their population is declining. They regulate the whole ecosystem balance. That's why they are important. Look at the snow leopard, leopards, tigers, lions, a lot of threats are available across the range countries through the illegal hunting, commercial trade happening, that is also illegal, poaching. So all those concerns are there. And these long-ranging species, so these long-ranging species needs a lot of efforts at intergovernmental level because their distribution is overlapped around different countries. So there, as I mentioned you before, there are different kind of the molecular markers or the genetic markers that can be used, but microsatellite markers are best suited to identify the recent changes in a population or a species. So in genetics, even 100 years is almost like a recent change. So we try to see how potential are microsatellite for species conservation. So first you can identify an individual like a DNA fingerprinting. If you use like, there are some markers that can be used for the species identification also. Once you have a genetic database available, you can identify the source of origin. You can identify the paternity, gene flow within the populations. These microsatellite markers can also be used for the captive enrichment, like captive breeding programs, identifying the suitable mate pairs among the individuals, and also nailing down the potential candidates for the reintroduction or the potential population for the reintroduction in the areas where these big cats had existed in the past. So there are a lot of benefits of the microsatellite markers. However, there are certain requirements also. Okay, so like generally microsatellite markers once required like species specific markers, although heterologous markers also work well, but you need to screen a lot of markers because as the species evolve, the primer binding regions bound to change across the species. Second, these markers are very costly because nowadays we are using the advanced genetic analyzers that analyze the fluorescently labeled markers. So these markers even costly. Third, the data requires a lot of technical expertise in terms of handling. All these things are some kind of hurdles of adoption. So with this project, we are trying to assist the wildlife managers, equip them with the technology so that they can use it like for a routine population monitoring. Like for an example, nowadays, forest department across different range countries, they are well equipped with the camera and camera trap technology. So they can do it on their own. They don't require any intervention or support from the academics or research institutions. So daily or day-to-day management of a population as a responsibility of our wildlife managers if they are suitably equipped with this technology, then they can use genetics also for the. So our idea was like make a technology which is cost effective, time efficient, and simple, and then train the people, distribute the resources across the range countries, specifically with the park managers. For that, we need a unified methodology. To make it a simple, we need some microsatellite markers pool that are accepted across the landscape by different institutions and that are working across the species. Like for many species, you can have different set of microsatellite markers for DNA fingerprinting, but the issues which are related to the management of big cats, to address them, you need to have a unified methodology. I'll try to explain you why. Like, for example, one of the biggest cause of or the potential threat to the survival of big cats is the illegal hunting and trade. Traded articles generally don't have any identification features. So many times they are mixed. Sometimes you have a lot of problems in identifying the species, so many things. So we want, like, if animals are traded across the countries, then every time if they identify tiger or a leopard or a snow leopard, for an example, or a lion, then they need a different set of the markers. To overcome this, we are trying to establish, or we propose, like, let's have a one methodology common. Till now, within India itself, you don't find a common agreement over adapting a single unified methodology, if in fact for the tiger itself. But in this project, we are trying to establish a unified methodology across the big cats. So we started this project in 2015. So first we tried to develop the unified methodology. So we started the mining of microsatellite markers from the whole genome sequences. So what we did is, like, we sequenced the genomes of tiger, leopard, lion, several individuals, and several subspecies, and then align these genomes to one another, and then find it out the consensus regions, and then design the primers for these regions, so that these primers will amplify equally among the different species. So this makes the primers very robust. And then after making these primers, we tried to test them. So initially we had a potential loci approximately 100. We filtered them, and finally now we have like 32 markers. These markers have a uniform amplification success rate across the different species. We published this article in scientific reports. So the current methodology includes the 8 different multiplex PCRs with a high efficiency rate, and these are the sequences of these primers. And if you look into their naming, like this A, B, A, C, whatever is written D, this all represents the different chromosome level. So we map them and we try to make like genome-wide coverage also must be provided by these markers, so that whatever the information we are getting with this marker actually represent the entire genome information. So with amplifying only 32 markers, one can get a lot of more information about the overall health of the particular species or a population. So first time we applied this methodology in Land of the Leopard National Park to study the Amur leopard population. So this study we started in 2019. Before that, we made an MOU with the Russian authorities. We provide them the training. And after that, we started the joint research. So Russians were involved in the experiments as well as field sample collection. We also went there, some Korean students went there, and then we collected the samples. Very simple way, we just identified for species, then gender, then we did the DNA fingerprinting. Also, we try to amplify the mitochondrial DNA sequence to identify the maternal lineages, because we know this population had a very low genetic diversity based on the previous results, and also the information that is available, like the 100 current population size of 100, in 1990s, the population was less than 20. So 5-fold increase has happened in past 20 years or 30 years. So this population must be having signs of inbreeding, we suspected. So we amplified the mitochondrial regions also. Also, our partners in Russia, they were keen in some kind of information like how much old sample is suitable for DNA extraction or better results, because resource is one of the limitation to conduct a study for a long-term monitoring. So we want to provide them some information like, is the technique efficient if you are using approximately 4 weeks, 6 weeks, or 8 weeks old samples or not, something like that. So we collected almost like 350 samples. Majority of them were tigers. And these are the numbers of the different seasons these samples were collected. So we participated from 2018 onwards. Before that, these samples were collected by the Russians, the researchers, and all were included in our study. So Land of the Leopard National Park, which is here, it has a leopard population as well as a tiger population. So tiger population is very less, almost like 40 individuals are surviving among tigers, and leopards are almost like 100. But when we look into our samples, what we got is like, it was like multifold increase or the tiger samples compared to the leopard samples, which was very unexpected for us. Then we look into our sex results. So we find like majority of the samples belongs to the males. Then we inquire to the Russian researchers, those who are involved in the sample collections. We asked them what might be the reason. So they said they collected everything what was available there. Then we asked what roads and tracks you followed. They said they followed the main major roads because this area is very long. So they used the car and then collected the samples whichever they encounter nearby the roads. Then we get some idea like why why our results are like that, because major roads are generally used by the tigers, because leopards live near the cliffs or in the mountains, and those regions were less frequently visited by the Russian researchers, those who are collecting the samples. Secondly, more male samples. Again, one of the reason is like they are collecting like major roads. So in major roads, for the territorial markings, the animals, male animals do more. So we got more samples. The another question like we asked like, so when the field researchers collected the samples, we asked them, okay, you just identified these species because they are experienced enough. And we try to compare these results with the genetic method species identification. Our results suggest that approximately 90% of the times, the species identification by the field researcher as well is similar to the genetic finding. So in future, if they want to conduct a genetic analysis, we recommend if they can do the genetic species identification, that's most welcome. But otherwise, even without that, they can directly go for the microsatellite genotyping to save the resources, because 90% of the time they are pretty sure based on the scat morphology which species it belongs to. So this is the distribution of the leopard individuals in the Land of the Leopard area. So total 24 leopard individuals were identified. And we recommended like only 12 microsatellites out of the 32 microsatellites for individual identification. And remaining microsatellites can be amplified for the genetic diversity assessment. And these microsatellites were amplified in 3 multiplex PCRs with high amplification success rate. So that also makes the overall reduction in the cost of the genetic analysis. We found like the samples that were collected between 2 to 4 weeks, they had a pretty high amplification success compared to the samples that are more than 4 weeks old. This is true in case of Russia because that landscape has a very low temperature, almost like a snow in most of the time, but may not be suitable for other species or other landscapes. Genetic diversity was found like with a heterozygosity of 35% and mean number of alleles of 2.5. So this genetic diversity was found less compared to the other studies that were published before. Similarly, in the mitochondrial DNA diversity, we found 2 different haplotypes with only 1 point mutation. In past, there were like 4 different haplotypes were found, and out of these, 3 haplotypes were not seen in our current genetic assessment. So what we assume is like either these population during the course of inbreeding has lost these maternal lineages, or still we need more samples to capture these. So we are continuing this genetic monitoring. So until 2022, we were able to collect these samples and the results are very much promising. So we'll come out with the results very soon. Genetic bottleneck was not detected. So, but this also needs to be reinvestigated because more number of males were there. So male bias population monitor sample collection might have resulted in some kind of an altered results, because here you have a loss of maternal lineage, but at a microsatellite level, we are not detecting the bottleneck signature. So overall, this collaborative research was found useful for the Russian counterparts, and they have adopted in a regular way for monitoring the tigers and leopards in the Land of the Leopard National Park. So we also try to compare it with the other studies. That's okay. So we published this our research in PLOS One. Currently, we are working on one another project. It's about the Sundarbans tigers in Bangladesh. So this project is supported by the Ministry of Environment, South Korea, World Bank, Bangladesh Forest Department, and CGRB at Seoul National University. So in this project, we are providing the information, technical cooperation, and also assisting them in the sample collection and genetic analysis. But every work is undertaken by the Bangladeshi researchers, either partially or fully within the Bangladesh. So our aim is to equip them to make them independent to conduct genetic analysis. So these are the project objectives. We are just looking into how the population is fragmented, how the gene flow is happening between the different parts of the Sundarban, etc. So in last year, we started the sampling. We covered the entire Sundarban region within like 10 days. So we had a team of more than 20 researchers with two different boards, one big shape. So we covered. In Sundarban, there is a one big problem, like because it's a mangrove forest, a lot during the tides, it's very difficult to get the scat samples, which are the main source of DNA extraction. So rather than focusing on the scat samples, we try to collect the hair samples. So this is one of the unique way to collect the more samples from a landscape like Sundarbans. So we just follow the tiger track approximately 50 meters once we get off from the boat, and then look inside the leaves where the animal has moved and left the hairs. So we collected like more than 150 samples, and then we identified the species of these samples. So this is the pug mark of tiger. So like my field researchers, they're going there, and they had their lab, so they are doing the lab genetic research in the Bangladesh. Also, we are providing a lot of assistance in getting good quality DNA out of the museum samples and the forensic case investigation, so that they can identify the source of origin. Because if you see this tiger or many other tiger skins, if they open, it doesn't look like a Sundarban tigers, because Sundarban tiger is approximately half of the size of the mainland tiger. But these are, so from where these tigers are coming, no one is sure about it, because a lot of illegal activities are happening in Bangladesh. Smuggling is happening for from third countries. So we need to look into all these things. So we are just working on it. So maybe by the end of this year, some results will come out, and then we'll share it. Thank you. Any questions? So most welcome.
Anyone, any question? Thank you, Dr. Pandey, for presenting very simply how basically now main genetics tool we are using for the wildlife management. So generally genomics people are using for looking the academic purposes, but generally if you see the real-time management, real-time monitoring of animal, which is beneficial for the conservation. So this kind of research is very important, and especially your cooperative work with Bangladesh, Korea, and Russia. That is very, I think that is good way to do science and research so that we can generate some good data. So
Yeah.
thank you for your presentation.
Hello.
So, yes.
Hi. This is Rita Sharma. Hi, Puneet.
Yeah,
Hi, Rita.
Can you hear me?
Rita.
Yeah, I can hear you. Hi, Rita.
Okay, great. This is great. I heard your talk and this is really great. I'm really impressed with the kind of good work you're doing. Just a small question that I have about this Sundarban tigers, because I recall from Dr. Sujit's last work on these tigers. So this is an island population. And as you mentioned that the tigers are really, dwarf. I mean, they are smaller than the mainland, right? So the thing is that, are you planning to look at their adaptation as well? Adaptation to this special environment ecosystem?
Every future research will depend on the requirement of Bangladesh Forest Department.
Yes, that's true.
These samples belongs to them, the facilities only. I'm just supporting them to make them
Okay.
if they want it. They are most welcome. We have a lot of whole genome sequencing facilities, everything we have. But only thing is, we need a willingness from their side. We have a resources to spend, no issues. But till now, they just want to have some population estimates because their population estimates are really some kind of problematic. So they want
really?
something because camera trapping you cannot do in Sundarban. So only thing is like they identified based on the pug marks. Even in that landscape, identifying tigers based on the pug mark is very difficult.
Yeah.
So that's a big issue.
Okay. Thank you.
Thank you.
Okay, Dr. Pandey, for your presentation. And I think this is time for a lunch here also.
Okay.
So we will bind our talk here, and remaining talks will be after lunch. So I think we will again join the this webinar at
Sujit, I just I'll ask Dr. S.P. Singh, who's basically our domain head, to say about a few words about the today's presentation.
Sir is online. Okay.
Yes, sir is online.
Sir, can you give me some words about today's talk, whatever, because you are discussing, you are looking our talks, whatever 4 or 5 talks we had. So can you give some word?
Okay. My sincere thanks, and I was really exciting seeing the Puneet presentation because he has worked with us in Amethi. So congratulations lot to Dr. Puneet and other also, which I have also gone through them.
Thank you, sir.
Very informative presentation up there. And Puneet is looking very bright now, more than that. And we wish that he should come whenever he visit to this site. Really, the area which he has taken, that is the most requirement as on today in that this canine family. We know that still there are a lot of gaps there, and phylogenetic is very, very important because now things has changed. We are not isolating our population and we are migrating, translocation, all that is happening. So if we do a translocation, keeping view of this phylogenetic value, that will be creating a more better genetic diversity. So his research is so pertinent, and now this is coming up very nicely. Technique has come up, and the forest department can really very thankful the scientists like Dr. Puneet for this type of study for future avoiding any kind of genetic drift or so much, because the way our habitat is getting isolated and pocket wise, we have, if we keep on that population, that particular protected area for a very, very, very long, possibly there will be a genetic drift and genetic loss. The another dimension has to be added. that is translocation, transfer animal from one PH to another for more genetic diversity. Only then we were able to survive these animals in a very long terms. This is my observation. Thank you very much, Dr. Sujit and Randeep Singh for inviting such an outstanding officers, the experts who are talking about. This is also new technology and is directly and indirectly related with green technology. Carnivores are on the top. We have a Project Tiger thing, total ecosystem that we are taking care, but not only carnivores, we are taking as a whole for all species. And thanks to the Project Tiger, that they have not saved only the tiger, but they could able to save many species of vegetation, flora and fauna, which has become totally safe and they are no more endangered. Thank you very much to everybody. Thank you very much.
So
Sir,
after the lunch.
Rita ma'am is here. So ma'am, I think you should wait for some time, because after lunch, then again we will join and we start your presentation.
No problem. I'll finish my coffee till then.
Okay, okay. I think it's morning time in Germany.
Hi, Ranbir. Yes, it's morning. It's 10.
10. Okay.
How are you?
Fine.
Good.
Okay, ma'am.
Go
So we
ahead,
can go for lunch, then we can start again.
Okay. All right. Thank you.
Okay, all the participants, we'll go for lunch, and then we will again join this meeting at 2:15. So we have almost half an hour, break, and after that we'll join at 2:15. So I am sharing this thing.
Do we have to leave the stage?
No, ma'am, no problem. You can. So I'm just.
Okay.
Yeah.
So now we can start, sir. Yeah. So very good afternoon, everyone. Now this is post-lunch session, and in post-lunch session we have two talk. One is by Dr. Rita Sharma and another is by Dr. Bhimda Joshi. So now we will start with Rita Sharma. Dr. Rita Sharma is going to present about the assessing the suitability of genome-wide SNP markers for conservation of endangered Borneo elephant. So before starting her talk, I would like to say something about Dr. Rita Sharma. So Dr. Rita Sharma is one of the few among the few researchers who started genetics work in India, especially on tiger conservation. So I think she was first or second student who did PhD on tiger by using genetic tools. For her PhD, she received DART fellowship, and she did her PhD in University of Potsdam, Germany. And after that, she joined her postdoc in Portugal, and she worked for elephant conservation. And now she is affiliated with the Wildlife Institute of India, and she is also working there in affiliation with the Wildlife Institute of India. She is working for elephant conservation there. And for her today's talk titled Assessing the Suitability of Genome-Wide SNP Markers for the Conservation of Endangered Borneo Elephant. So I would like to invite Dr. Rita Sharma for her talk. So ma'am, please, you can start your talk.
Thank you, Dr. Sujit Singh. Thank you so much for the great introduction. And now I share my screen. Can you see here now? Yes.
Yes, ma'am. It is visible.
Great. All right. So
it is visible, ma'am.
Sorry, there's something coming over here. Okay, great. So anyway, good afternoon, everybody. And I'm Rita Sharma, as Dr. Sujit Singh has already said some good words about me. So I'll talk about in this session about my previous postdoc work on Borneo population and conservation genetics work on Borneo elephants. So, but before I start my talk, I would like to thank the organizers at the MIT Institute, particularly Dr. Sujit Singh, Dr. Randeep, who invited me to present my work on the occasion of faculty development plan on green technology for sustainable development. So I'm going to discuss today about the different markers types. I mean, what are the different markers that we use for the genetic studies for the conservation of different species. So first, I will tell you about I'll tell you in brief about the Borneo elephant, and thereafter I'll go into more details about different markers and how are we going to use them, and how did we use, and what are the benefits and advantages of different types of markers that are available for the genetic studies. For example, the microsatellite markers and single nucleotide polymorphism, that is the SNP. Okay. So the Asian elephant, as you see here, their distribution. So they once ranged from the Mesopotamia in the west across the Indian subcontinent to the Southeast Asia, including the islands of Java, Sumatra, and Borneo, and they were also present in China. But due to recent habitat loss and fragmentation, it has been extirpated from approximately 95% of its historical range, and they exist only now in a number of fragmented and small isolated populations in South and Southeast Asia. So elephants are basically mega herbivores with large body size. So they are really, really susceptible to human impact due to their long generation time and slow reproduction. And due to this recent scenario, they are categorized as endangered according to the International Union of Conservation of Nature. So currently there are 3 different species of Bornean elephants across their range distribution in Asia. So the first is the Indian elephant that is present in India and other Asian mainland. The second is Sri Lanka, which is also endangered. And the third one is Sumatra elephant, which are just present in the Indonesian island of Sumatra and which are really critically endangered. And they are even, the numbers of Sumatra elephants are even less than the Borneo elephants. So for Borneo elephant, you see that they're just like 2,000 individuals left in the island of Borneo. And so the Borneo elephant recently is identified as the 4th subspecies, and I'll tell you why later. So as you can see here, these Borneo elephant, they are smaller than the other mainland species and also from the African main species. And they are also known as pygmy elephant, and they have a very different behavior. And the morphology is also completely different from the other mainland species, such as their small height, straight tusk, as you see here, and the tail is also very straight. So according to a previous study, the Borneo elephant, they separated from their most common ancestors around 300,000 years ago. So now coming back to Borneo again. So if you see here, the Borneo is the 3rd largest island of the world, and about half of the Borneo natural forest have been lost between 1950 and 2020. So Borneo lost an average of around 850,000 hectares of forest every year, and the forest cover has dropped approximately 1/3 from the original in 1950. And these Borneo elephants have a very limited or restricted distribution. They are just restricted to the 5% of the island and the extreme northeast of Borneo, and mostly in the Malaysian state of Sabah. So Sabah is also highly fragmented. The elephant habitat, as you see here in the pictures, the habitat is completely fragmented. And the main threat is, as with all the other mainland Asian elephants, is habitat loss and fragmentation. But here the fragmentation occurs through this conversion of these existing forests to commercial plantations, such as this oil palm, or for permanent human settlements around here. If you see here, for instance, in the last 50 years, 80% of these forest in the lower Kinabatangan, that this is Kinabatangan River. So around 80% of the forest has been converted to agriculture land and for human settlements. So just to let you know, what are the other large mammals present in Borneo? Borneo is highly diverse and species richness due to it's famous for species richness and their density. And the thing is that other large mammals that are present in Borneo, for example, this is proboscis monkey, orangutan, and this is banteng, or also known as tembadu. And the large mammal is this Borneo or Sumatra rhino, which has recently gone extinct in the, I think in the last couple of years. So the questions that we had in our project is, can we detect the effect of habitat fragmentation on the level of genetic diversity of Bornean elephant? And can we estimate the genetic diversity and its distribution across the entire range? So we just performed this study to determine these effects and also to detect possible genetic differentiation between the local populations using a large sample size from across much of the elephant range. And we were also interested to know the past population history of the elephants in Borneo. I will show you later I'll tell you more about the controversial theories on how the elephants they came to Borneo and what is the origin, and with the help of these genetic tools, how did we solve this mystery? So to carry out these population genetic and conservation study, we need markers. As Dr. Puneet Pandey has already told you so many things about these genetic markers, how we can use, what are the benefits, what are the advantages of different markers such as microsatellites or SNPs. So the thing is that for any kind of marker that we are going to apply on our study, there are certain criteria to select these useful markers. For instance, the marker should be polymorphic because they have to be informative, right? So, and then they should only amplify when you apply the markers to the samples, they should only amplify a single locus, because so that they occur in only one location in the genome, and there is no ambiguity or confusion about their number or position. And the markers should be also located on different chromosomes, so that they are independent, and then you can make more assumptions and calculate the summary statistics without any bias. For some applications, for instance, the study of population size and the demographic change over time, the marker has to be neutral, because otherwise, and they should not be, they are not correlated with selection or adaptation, because otherwise the selection or adaptation will confound the estimates of the population size parameters. But if you really want to study selection or adaptation, then you can use the genetic the locus that you presume to be under selection for a particular species or for population. So we already had some microsatellite data for the Bornean elephant, but we decided to go for some genomic markers, because there was this transition was going on from microsatellites to the single nucleotide polymorphism markers. And we thought that, okay, we also isolate few SNP markers for the Bornean elephant, and then we see how the data looks like. And why single nucleotide polymorphism? Because they have considerable advantages compared to the more traditionally applied microsatellite markers due to the following reasons. Because they are common throughout the genome of most of the species. They occur about once, like for example, in case of human genome, they occur about once every 500 to 1000 base pair. And they can be used like any other genotyping marker, like microsatellites. But then here, in case of SNPs, they are biallelic. They're just 2 alleles at this point. So you need more loci typically. So the thing is that if they are biallelic, and in case of microsatellites, they are polymorphic, right? They have multiple alleles. So the thing is that if you use microsatellites, you will see the genetic diversity is high, but then you will not reach that kind of resolution with SNPs. So therefore, as I said, that you need more number of SNPs than the microsatellites. Also, in case of SNPs, they have a well-defined mutation model. However, in case of microsatellites, it's complicated and it's not always very easy to know the mutation model behind those alleles. And also, as Dr. Puneet Pandey has mentioned, that for microsatellite, it is very difficult to calibrate them across different laboratories, and sometimes the data is very difficult to trust because they have used different machines and their laboratory conditions are different. So, but in case of SNPs, you don't require calibration across different laboratories, and it's very easy to collaborate. And the genotype calls are straightforward, and the biallelic nature of the marker leaves little room for subjective scoring decisions. And also the SNPs are very easy and suitable for the amplification of fragmented DNA from non-invasively collected samples such as dung, hair, or your forensic samples, or historical material like bones, etc. So to start the work, we did some sampling. So for the sampling for the genomic markers, to develop the genomic markers, you need really good and fresh samples. So that has to be the blood. So what we did is that we collected some blood samples from the captive individuals in the wild, and we obtained something like 8-9 samples, fresh blood samples of the elephants. And the second thing that we did, we sampled some wild individuals. So in Sabah, this is the map of the Sabah. Here you see the elephants are distributed in 4 different ranges. So we collected the samples from all these 4 ranges, which is like Lower Kinabatangan, North Kinabatangan, Central Forest. This is Central Forest, Tabin here. So we collected all these dung samples for the wild individuals. And you see how this is the biggest poo I have ever seen in my life, elephant poo. So in total, we collected around 250 individuals from different forest fragments that represent continued and fragmented landscape both. So to develop these genome-wide markers that are the SNPs, we used 2 different techniques. So that first one is the rat sequencing, and the second one is 454 shotgun sequencing. And we used these methods way back in 2011-12 when the genome sequencing technology was still in its infancy and super expensive. So what we did is that we actually extracted the DNA from all this blood and the fecal dung samples, and then we developed the markers using these 2 genomic method, and then we applied the marker directly on the samples. So I'll just briefly mention the method because the details are already there in the literature. So what we did is the first is the RAT-seq technique, which is known as like restriction site associated DNA sequencing technology, in which what we do is that we digest the genomic DNA using different restriction enzymes, and we digest the whole genome, the DNA from the blood, and then we apply the enzyme, and then we digest the DNA, and then what we get in return, thousands of reads, and which are next to the restriction site, And then you have many samples, you have many genome. So you have these enzymes that will digest the genome at different points, depends whether the enzyme site is there or not. And then what happens is that the technique, what it does, it ligates adapters to the both side of the fragment. And this adapter contains an amplification site, a priming site, and a barcode for the sample ID. And then the adapter just sticks to the restriction site. And after that, you do more sequencing, and then you have a lot of reads, and then you assemble the reads, make a contig, and then you align a lot of contigs together, and you identify the SNPs based on that. And you can also just when you have these contigs ready, you can map it to the reference genome, and based on the comparison with the reference genome. For instance, in this case, I think we used the reference genome of this African elephant, and then we identified the SNPs, the polymorphic positions of our elephants in the genome of our elephants, and compared to the African elephant genome. So the second technique is the 454, in which here you see the genome is just fragmented and sheared and sequenced many times, and then you align the reads together and build the contigs, and many thousands of sequence reads are obtained through shotgun sequencing, and then overlapping sequences are identified and aligned computationally to build the contigs, and these contigs are then compared and aligned, and the SNPs are identified again in comparison with the reference genome or without a reference genome. So here in this case, we just use 2 individuals, but in case of rat sequencing, we use around 8 individuals. So these are just some of the summary statistics that we obtained through this whole genome sequencing thing. So way back then, the technique was not that evolved. No, it was very old, and now the sequencing technology has changed a lot and evolved since then. And you have these double digestion rat sequencing. Before it was just a single enzyme. Now they use 2 enzymes together to have more number of reads and better position of the SNPs, etc. So here, what you see that using both the methods, we identified several SNPs, thousands of SNPs, and the results are not that comparable because we use different number of individuals and different methods. But I'm just highlighting the summary statistics and the number of markers that we have developed using this high throughput sequencing approach. And after that, we sorted out some SNPs, and then we identified some which are suitable for the sequenom assay design that we used later to genotype the SNPs. I'll tell you just, I'll mention that later. And so you can see that how these numbers, they are quite different for the different method. So we designed a group of multiplex genotyping assay using the sequenom mass array design software. And we used a subset of SNPs for validation on the elephant blood and dung samples. So once the assays were identified, we carried out large-scale survey of their frequency using the sequenom IPlex genotyping platform. And we genotyped around 194 SNPs on blood and dung sample. And this assay design was used for genotyping in a 384 well plate that included diluted DNA samples from blood and dung samples. So our genotyping data was not that good in the sense because the Borneo elephant has a low heterozygosity. So we found that around 67% of the SNPs were monomorphic, and monomorphic means you have just one single base, but around 31%, only 31% of the loci were polymorphic in both fecal and blood samples. So we repeated the genotyping for each sample around 4 times, and we created a consensus. And we also found that the genotyping error rate across the sample was high, not that low, but they were similar when we compared it with the microsatellite data set. So we repeated genotyping for each sample for around 4 times to get this genotyping error rate. So here, this is just the comparison among the markers, that is SNPs and the microsatellites. And we compare the measures of the population genetic diversity, like the expected and observed heterozygosity in our SNP data, with the same samples that we genotype using the microsatellite markers. So what is HE? So HE is the gene variability of a population, and it is usually based on allele frequency. And HO is the number of observed heterozygotes in the population. And based on these numbers, you can calculate the genetic diversity of the individual as well as the population of the species, so and so on. So both data set, if you see, they show that Borneo elephant has a low genetic diversity, and the diversity measures were low for the SNPs than the microsatellites. And I will tell you later why. So, but also we compared it to the previous study done by Fernando, and they also used just 5 microsatellite loci and genotype only 15 individuals, and they also found very low genetic diversity. So you see at the average HO on the y-axis and average HE on the x-axis, and you see that this the genetic diversity of the microsatellite versus SNPs, and then the other the data from the other study. So what the interesting part of this study was that we found overall concordance among the SNPs and microsatellites in the amount of genetic variation within the elephant population. That is, for example, see the Deramakot and the Lower Kinabatangan, they have really high levels of genetic diversity compared to the other populations here. So this is the SNP data here, HE for the SNP, and this is for microsatellite. And this is observed heterozygosity for the SNPs, and this is for the microsatellites. So you see these two populations have higher genetic diversity than the rest of the populations. And as I said before, the genetic diversity was low in Bornean elephants compared to the other Asian elephant populations. Also, when we compared this population structure, so we found that the Bayesian cluster analysis supported the existence of 2 weak genetic clusters, and they grouped the individuals from the Lower Kinabatangan and Deramakot, as I showed before, with high genetic diversity in one cluster, and the rest of the individuals from other population in another cluster. So we found a considerable level of genetic differentiation between the two populations using the FST values, and this shows that there is a limited gene flow between these populations. And also, when we looked into the social structure and the samples that were collected within the social group of the elephants, we found that the average relatedness within the site was lower than the samples that were collected within the family units. So anyway, the average relatedness was quite high in both the data set using the SNPs and microsatellite data. So we also identified few microsatellites from the RAT-seq technique and the 454 technique, and we identified the following di, tri, and tetra microsatellites. And out of the 55 potential microsatellites, we found that the 38 microsatellites, they produce these scoreable peaks. However, they were all found to be monomorphic across all the Bornean elephant samples. But then we found that 5 out of these 38 were polymorphic in another Asian elephant subspecies, even though we had just 3 individuals. And we also identified few loci with fixed heterozygosity. So coming back to the origin of elephant. So we use the same data set to understand the origin of elephants on Borneo, so which has been very controversial. So there are 2 competing hypotheses that have been proposed. First hypothesis is recent introduction, and the introduction hypothesis is based on historical records suggesting that the current Bornean elephant population represents the descendants of a domesticated herd that formerly existed on Sulu Island, which is here in Philippines. And they were introduced to Eastern Sabah by the Sultan of Sulu in the 17th century, which is like 300 or 350 years ago. And these historical records also suggest that the original founders of these elephants in Sulu, they came from the Java elephant population. And it was also reported that only 2 elephants were introduced to Zulu in the late 13th century. And the second hypothesis is ancient colonization, which argues that the Borneo elephant is indigenous to Borneo and the wild population was not introduced by humans. So on that basis, it was suggested that the extinct Bornean elephants are derived from the endemic or Pleistocene progenitors. So what we did to understand the origin of elephants on Borneo, we used ABC, that is Approximate Bayesian Computational Tool, to reconstruct the demographic history of elephants on Borneo. So what ABC does, it makes use of the simulations to contrast the complex demographic models that are commonly used. So we use, so what we did, we actually kept the demographic scenarios very simple, and we tested 5 simple and 2 complex models in total. So I don't show here the complex scenarios, and only they will discuss the simple ones. So the model, they just, they differ from each other on the date of introduction and on whether the founder event occurred. So the first is instantaneous decline, where the large ancestral population that underwent instantaneous decline to their current stable effective size population without a founder event. Then second is the exponential decline. The decline is exponential. And the third is ancient colonization, where the large ancestral population decrease as a result of an ancient founder event, and then it grows exponentially to the present size population. The fourth is a recent introduction, as I mentioned before, the recent introduction hypothesis, where the elephants were introduced from Sulu or Java with a recent founder event, and following which the population experienced a demographic expansion. And this one is the 2 introductions, where we combine both the AC, ancient colonization, and recent introduction models together. One ancient from the mainland to Java, and then from Java to Sabah. So for all the ABC analysis, we used a total of 6 summary stats. And what we observed that for all these models, the AC model, it received a strongest support, which means that it had a high posterior probability, as you see here. So the posterior probabilities clearly rejected models assuming a recent introduction from Sulu and Java, as well as the models with no founder event. This model assumes that an ancient colonization of elephants from into Borneo, it accompanied by a founder event, and the models emulating a recent introduction and 2 introduction had a very low posterior probability. Although our results do not enable us to pinpoint the precise time of colonization, but we find that the best fitting scenario, that is the ancient colonization, involves an end of Pleistocene bottleneck, probably around, you could say, 11,000 to 18,000 years ago, roughly coinciding with the end of the last glacial maxima. So we think that it is possible that elephants could have dispersed naturally from Java to Borneo rather than this mainland Asia, when the sea level was low here, around at least 40 meters lower than at present. And these major Sunda islands, Java, Sumatra, and Borneo, they were connected several times during the Pleistocene via land bridges during the periods of low sea levels in the late Pliocene and Pleistocene, until the end of last glacial maxima around 15,000 years ago. So here we are, the last slide. So we conclude from this study, we concluded a number of, we noted a number of facts, and some of these are like, we observed that high number of SNPs can be identified even in a genetically depleted species like the Bornean elephant, and you can use these SNPs to study the population and conservation genetic study of any endangered species. And we noticed also that the rate of validation assay was really high for the 454 than the RAD technique. It's because in 454 you have the context size is bigger than the red, so you can identify more SNPs in a bigger sequence than in a shorter one. And similarly, we found that the SNP are because the SNP are generally biallelic, so as a result we require more SNPs, like 2 to 6 times more SNPs are theoretically required to match the resolution of microsatellites. And studies have also shown that the significant power to detect population structure at a very recent divergence time required as many as 15 times more SNPs than the microsatellites, and to obtain a very fine resolution of population structure. But this study was done a long time ago, so we didn't have so much high coverage, and that's why we also had problems such as fixed heterozygosity. And at the end, but this study reveals that the genetic diversity is unequally distributed between the elephant populations in Sabah, and it is really important to reestablish and restore the habitat connectivity between all these elephant populations. And the conservation should really focus to, maintain the population connectivity between these individual populations. So thank you so much for listening to this talk. And I would really appreciate if you have any questions. I'm happy to give you my answer.
Participants, any question? If you have any question, you can ask. Or if you also can type a question here in the chat box, so I can ask. Okay, ma'am. So I have one question. You have used both marker like microsatellite and SNP, both one. So and both have possibility of error. So chance of error is very common because we are working with non-invasive samples. So which one is best if we see the error?
Which one is the best, you mean?
Best to use.
Okay, according to my experience, yes, I think SNPs are better, because the thing is that in SNPs you can have a really small fragment size, like less than 100. You can use 90 base pairs, 60 base pairs. I think in microsatellites you cannot go down so much, right?
Yeah, it's for few markers we have, but error rate is very high for those markers. But
exactly.
Yeah.
And because microsatellites are, polymorphic, they have multiple alleles. So I think in SNPs, it's not the case. You will just miss 1 or 2 alleles, because it's just, these platforms, there are so many genotyping platforms. So what they do is that they identify the marker based on their density. So if it's A, G, C, T, so there are 4 different bases, right? So there's a different density for these alleles, and is the way that they, call the alleles, call the genotypes. And that way you will have less genotypic error in case of SNPs than microsatellites. For microsatellite, we use the length, right? The allele length.
Yes. And the calling is another problem. If you are not very much precise, very much expert, then that is another problem with microsatellites.
Yeah, I know. I still struggle with sometimes microsatellites. Calling genotype is not easy.
Yes,
it's so difficult.
If you are not, used to, at least you need to have each and everything, then you can, start calling.
Yeah, and also if you take a break, for example, if you don't analyze a microsatellite for like 2 months or 3 months, and then you come back again, you forget.
You need to recall everything. Yes, this is the main thing.
It happens to me.
Microsatellite.
It's complicated.
Yes.
The SNP is more automated,
Yes, ma'am. So actually, density is also high, I think. Repeats and all these things.
Yeah, this is good. And also another thing that I noticed in my project is that sometimes it happens that you have 2-3 SNPs. So you have a SNP and then you have this primer binding site. So what happens is that sometimes you will have a lot of SNPs together. So then you cannot, design the primer. So then you have to discard a high number of SNPs. So, but if you have thousands of SNPs, you can identify at least 100, 200. That is not a big problem, I think.
Yes, because in microset, if you have 10, 12 marker, then excluding 1 or 2 markers is a little bit difficult, because that will reduce the diversity and all these things. Yes.
Exactly. That's true.
So, okay, ma'am, I think there is no question here. So I think, ma'am, we will go for the next talk. So thank you for your presentation, ma'am, and you devoted time. I think the this time was little bit very different there in Germany. It was in morning because for me it was very difficult to accommodate you, Dr. Puneet and Dr. Yoni. Everyone is telling I will go after lunch, and in morning I was unable to. Then I skip. I you know. Give my presentation in morning so that I can accommodate you all there.
Thank you so much. Thank you so much,
Thank you, ma'am.
Dr.
Thank you,
Sujit.
ma'am, for giving your talk.
Thank you.
So now we'll go for next speaker. I think Dr. Bhimda Joshi. So Joshi, okay, so Joshi already joined this things. So very good afternoon, Dr. Joshi.
Good
I think
afternoon,
you are in Indian zone and you are in Kolkata, so our time is almost same, so no problem with us. I think
yes,
because we had problem with Dr. Puneet. It was evening time in Korea. Ma'am was it was morning in Germany, so it was a little bit difficult for us. So Dr. Joshi, I think before starting Dr. Joshi talk, I would like to say something about Dr. Joshi. So Dr. Joshi is our very old colleague, and I think we spent last more than 10 years working in this field. And we worked together at Wildlife Staff India and Zoological Survey of India. So Dr. Joshi started his career in this field in 2010, I think. And then he shifted to Gurukul Kangri University for his PhD, but his association with WIA, it was still live. And he did his PhD in collaboration with Wildlife Staff Institute of India and Gurukul Kangri University. And after that, Dr. Joshi joined Zoological Survey of India as a postdoctoral fellow, and he did excellent job there. I think he spent 5 years there, and he's published more than 50 research papers, and all were very good. And in his group, Dr. Joshi, I think he was not direct supervisor, but I know very well that Dr. Joshi supervised almost 8 to 9 students, and all were now in end of year PhD, and they are submitting the PhD. So indirectly, Dr. Joshi supervised them in every aspect, lab aspect, personal aspect, emotional aspect. And I think Dr. Joshi spent very precious time there in Zoological Survey of India, and he did lots of job, good work there for the species conservation. So I would request Dr. Joshi to start his talk. Dr. Joshi, please.
So my slides are movable.
Yes, you can keep in slide share mode. Then I think yes.
Yes, I have.
Can you change the slide first?
Yes.
Yeah,
Is
now it is working. It is working.
Okay. Thank you, sir. Thank you for the introduction, and good evening, everyone. This talk was supposed to be given by the Dr. Mukesh Thakur, but due to some unavoidable commitments, he is unable to deliver this lecture this time. So I am here. So I'll be talking about the conservation genetics of mammals in the Indian Himalayan region. So that we work in the Indian Himalayan region since 2012. But in the I will talk most about what we did in the conservation genetics and genomics in the Indian Himalayan region since 2018. So this was the project funded by the National Mission on Himalayan Study, or to monitor the vertebrates in the Indian Himalayan region, where we work on the more than 30 species, but both for ecological aspects and the molecular aspects. Here I will mostly talk about the non-invasive genetics, and in these projects we collected about 5,000 samples of the mammals throughout the Himalaya. We have the study sites, 6 study sites covering from the western to the eastern Himalayas, having the 6 districts: Lahaul, Spiti, Uttarkasi, Sikkim, Darjeeling, West Kameng, and East Siang. We collected approximately 5,000 samples, sequenced about 37 mammals, generated 2,100 DNA barcode, used 103 microcetallosi, and genotype 13 mammals for the detailed population genetic analysis. So this was the basic one of the largest non-invasive genetic survey that we did, where we process about 4,800 non-invasive samples, in which 70% of the samples were found suitable for the DNA extraction. About 65% was sequenced, and in which 58% sequence were resulted to the wild animal, and that was about 2145 sequences, which resulted into the total 37 mammal species from the different area and the 44 species of the domestic animal, which is sympatric with the Indian Himalayan region. If we divide it into the species based on the Himalayas, so in the central Himalaya we got the 15 species on the DNA based analysis, 25 species in the eastern Himalaya, and 18 species in the western Himalaya. Those species which has a more than 50 samples, we process them for the detailed population genetic analysis, and we got around minimum samples of around some 53 snow leopard and 139 of the red panda. We process them for the detailed population genetic analysis with the 103 microset loci, and we found that we generated around 569 genotype out of these 1319 cells and 458 individuals. We characterized them for the detail, we generated the population genetic diversity of each species, and we got 2 to 14 haplotypes in these 13 species and about 15 to 60 unique individual in the each of the 13 species that we studied. So this is the basic distribution of those species which we have studied throughout the Himalaya, and we generated the population genetic account unbiased, which can be monitored in the interval of the certain time limits, which these generally it is assumed that these are the like the unbiased heterozygosity, nucleotide diversity, and effective population size that we calculate is not the sample biased. We also did the demographic analysis, and we found that most of the species shows the stable demographic history, but some of the species like Himalayan goral, red panda, Arunachal macaque, and common leopard has the recent decline in their population. This can be interpreted with the land use changes in the Indian Himalayan region about the 2.6 to 5.7 thousand years ago, which we can see the evidences of human presence in the Indian Himalayan region, and that led to the massive change in the land use pattern throughout the Himalaya. So that's the conservation implication of this study, was to map the spatial genetic diversity that, with respect to human mediated land use changes, and this information can be used for the effective conservation of that species. So Joe, what the PGA we develop here can be used for the genetic monitoring, long-term genetic monitoring of these species in the temporal scales. Further, we also address the taxonomic ambiguity that I will discuss in the later. Identified some of the ESU that can help to make the species and ESU oriented conservation plans in the Indian Himalayan region. So in terms of speciation, we did the whole genome sequence of Himalayan ibex, and so the Siberian ibex, basically, this Siberian ibex is distributed throughout the central and the northern Asia, including the Afghanistan, western and northern China, northwest India, southeastern Kazakhstan, and Kyrgyzstan, Tajikistan, and so Mongolia, and even south central Russia. So this species has a very wide distribution throughout this range. But when there was no genetic information were available on the Himalayan ibex, which is distributed in the small part of the India, basically in the Ladakh and the Trans-Himalayan range of the Himachal Pradesh. So first we did the genetic analysis using the short fragment of the mitochondrial cytochrome B, and we found that the ibex is distributed in the Indian part of the Himalayas. Trans-Himalayan range is showing the very distinct clade from the other that is distributed in the Kazakhstan and Mongolia and Russia. So we got one distinct pattern, but which although there was the report of different species present in the different subspecies present within this range, but what we observed was this distance was quite high than the subspecies level, and further we claim that this can be the evidences for the allopatric speciation in the Himalayan ibex. So we did three We did further we goes for the 3 whole genome analysis of the Himalayan ibex and compared it with the 3 genome of the 4 species of ibex from the entire distribution range. Then we did the phylogeny, population genetics, demographic history, molecular dating, and gene flow. And we found that our findings were consistent with our short fragment of the mitochondrial DNA, both using the nuclear variant of the whole genome sequencing as well as the complete mitochondrial genome, where the Indian Himalayan, the ibex distributed in the Indian Himalaya, specifically the in the Lahore and Ladakh and the up to the Tajikistan, that is forming the paraphyletic clade with respect to the other species, other ibex that is distributed in the other central and the central Asian landscape. So further, we did the demographic analysis. We found that Himalayan ibex has 2 major bottleneck events in the 0.8 to 0.5 million years ago, and another was the 50 to 30,000 years ago, with the intermediate large population expansion during the 0.2 to 0.16 million year ago. And this species was split from the about 0.1 million ago from the other Siberian ibex population. We also get the admixture and non-basin assignment in the species showed that Himalayan ibex is in the isolated population when compared with the other Siberian ibex population from the Tajikistan, Kazakhstan, China, from both sides of the China, and did not share any gene pool with its adjacent population in the any of the analysis, and shows that Himalayan ibex shared excess of the allele with the Tajikistan population and the Chinese ibex compared with its Kazakhstan population, which shows that the ibex distributed in the Indian Himalayan Trans-Himalayan range is the entirely different population. This can be due to the major geoclimatic events happened. Some of the valleys in the entire distribution range, especially from this area, becomes the wider at least 3 times deeper from since 3 million years ago to the present time, and likely one of the reason for the disjunct in the Pamir mountain region that may lead to the isolation of the Indian Himalayan population. So another study we did the exploring the Himalayan macaque in the Eastern Himalaya. In this Eastern Himalaya is the one of the biodiversity hotspot where the 5 species of the macaque distributed in the different areas, and the taxonomy of these species is completely unresolved. So what we did, this study is published in the by the Geological Survey of India, one of our colleague Abhijit Ghosh. He found that there are the Arunachal macaque which has been discovered in the 2015, which is found in the Tawang, and he collected some samples from the West Kaming district of the Arunachal Pradesh and found that this species is entirely different from the Arunachal macaque which is found in the Tawang. So the divergence time was reported for this species about 2 million years ago, and similarly the genetic divergence between the West Kaming population and the Tawang population of the Arunachal Pradesh was the higher than the 2 species Macaca tibetana and asamensis, which indicates that these 2 population may be the different species, and these results were consistently supported with the microsat loci, and which this landscape was entirely separated by the Sela Pass, is the one of the highest peak in the north in the western Arunachal Pradesh, and maybe the act as a geographic barrier for these 2 populations. So there are the some of the morphological differences were also observed between these 2 population of the West Kaming and the Sela macaque in the West Kaming and from the Tawang. So this study was published in the Molecular Phylogenetic and Evolution. Another was serendipitous discovery of the white chick macaque in the this in the Arunachal Pradesh, which is only found in the Tibetan Tibet region, and recently we also got the one record of this species. Further, there we also got the 2 chromosomal lineage in the white chick macaque, which indicates there are the male bias interrogation in this species. One, as we can see here, there were the mitochondrial genome, which is which is not showing any different genetic lineages. But when we compared it with the Y chromosome TSPY gene, there are the two that one sequence is going with the asamensis of the white chick macaque, and another is going with the macaca leucogenesis. So this is showing there are two different lineage in the same individuals, while they are not showing with the mitochondrial DNA. So this is subject to the further exploration of how these what are the evolutionary pattern of these species, and is there any hybridization between those macaque species in the north in the eastern Himalayas. We are exploring it further by the whole genome sequencing of five macaque species. Another interesting study was that the resolving taxonomic disparity in the genus Nemacolus. This is the Himalaya goral species, which has about 6 species distributed in the throughout the China, Myanmar, and the also the Bhutan and Indian Himalayan region. So we have the two questions that Mori et al. 2019 and Lee et al. 2020 question the existence of Chinese goral, which is the you can see here that angriescens distributed in the whole southern part of the China, south eastern part of the China, and this species they consider as that this is the Himalayan goral. So there is no existence of the Chinese goral, but they have not included any samples of Himalayan goral in the Indian Himalayan region, and that what inferences they have made based on the sequences available that is from the Chinese goral range, which is named as the Himalayan goral, but was collected from this range, and no samples were included from the Indian Himalayan region. Then we use the cytochrome B and control region markers to understand the taxonomy of this species, and we found that yes, there is the Himalayan goral, which has the very distinct cladistic pattern. This one very you can see this is forming the basal clade with the all goral species that is distributed to the eastward from this particular Indian Himalayan region. And yes, we found that no, there is the existence of this particular gray Chinese goral, which is quite different from the morphologically from this species. Another question was that the Himalayan goral assigned as a 2 subspecies in both western and the eastern Himalayas, but Welgen and the groups and Groves 2011 suggested the occurrence of 2 species, gray goral in the western Himalayas and brown goral in the central and the eastern Himalayas. So further we investigated that, we got some samples from the we did the camera trapping throughout the all Himalayas, and western Himalayas as well, central and the eastern Himalaya, and there are the quite morphological differences between all 3 goral species which is distributed. But samples we got from this area to this area and found that yes, there are there is no 2 species. There are divergence, but not the level of the species. So there are they can be considered as the 2 subspecies, Himalayan goral. So we concluded that from the Uttarakhand to the Sikkim, up to the Sikkim, there are 2 different clades, can be clades, and these 2 species, the clade number 6A and 6B, can be considered as the Himalayan goral and the Bedford, the 2 subspecies which has been which was initially assigned as the 2 subspecies, that is. So the Belgian 2011 and groups 2021 what suggested 2 subspecies, we told that this is the only 2 subspecies, not the species. So but this is further subject to the analysis of how these in the eastern part of the Arunachal Pradesh, how many other species are found. There are the report, there are some of the species may be present of the Himalayan goral. So this is the one table that we revised based on the different time of the study. So this species actually given, this species was given name as the here Grevensis, the Gracis was not considered, but here we consider it as a species, and these 2 subspecies of the goral in the Indian Himalayan region, and rest of the 5 species is also in the existence. So this was the basic study what we done, and there are so many others, but due to the time constraints, I would like to conclude my presentation here only. Thank you.
Thank you, Dr. Joshi. So anyone, if you have any question, you can ask. Rita ma'am, if you have any question, you can ask
Yes.
Joshi. Yes, yes, ma'am.
I have two questions. So the first is, what is the population status of the Himalayan goral? Because it was not clear in the slides, or maybe I missed something. I just want to know the details about the population size and yeah.
Oh, 1 second, ma'am.
And yes, congratulations. This is really great study. I am really happy to attend the talk.
Thank
here.
you. So, ma'am, as such, there is no population status of the Himalayan goral because no census has been taken so far. There are very limited studies going on. So we are doing the study on the Himalayan goral in the Uttarkashi and the Arunachal Pradesh also. But as such, there are very few studies that give the numbers, only the density 1.3 or 1.4 individuals per square kilometer, like that. So there is not exact estimation of the species because there is still the ambiguities in the species presence in the western and the eastern Himalayas. So very limited studies are available, especially they have given only the densities of about those species.
And have you covered the entire habitat of this species? For sampling.
Yes, sampling, representative sampling we have got from the Himachal Pradesh. We got from the Arunachal Pradesh also, some of the from the central Himalayan landscape. So representative sampling we have camera trap data also, what species, as you have seen that. But still those images which is showing the different code colors and the bottom of need to be still validated by the other markers, and there are more number of samples from that area, especially in the Arunachal Pradesh.
Okay. And the second very quick question. So you have shown the slides about, you have shown the details about the Himalayan ibex. Yes.
Yes.
And you say you have done some PSMC analysis, but I just want to know, have you done ROH analysis as well, the runs of homozygosity? Have you looked at the inbreeding status of the population?
Yeah, inbreeding status, because we have only 2 or 3 samples, but yes, we have run, but we did not get any inbreeding signature on that.
Okay, thank you.
Yeah, because ma'am, lots of population size is very large. If you see, in Himalayan region, especially in Ladakh and Laol Spiti region, this species is distributed across the landscape. So,
yeah.
But I think recently one student was doing PhD, I think. So she is doing some work. I think Joshi Gul is working something. So
Yes, sir.
She is doing PhD on conservation genetic aspects. So she will cover all the Ladakh region, Laol Spiti, and all these things. So she is, I think, about to submit this work.
Okay, great, good.
Okay, so any participant, if you have any question, then you can ask. Otherwise, I think this was last, talk of today's webinar. So I think after that we have one documentary hosting. So Randeep sir, I think if you host one documentary, whatever you have planned, so in between we will plan some question and all these things. So then I will share question with participants and then discuss.
Dear participants, today we are going to air a documentary about the reintroduction of the tigers from Ranthambore to the Saliska tiger. As you are aware that all the tigers, they have been finished by poaching in the Saliska Tiger Reserve in 2004. Then Ministry of Environment and Forest and the Rajasthan Forest Department, they have tried to repopulate the Saliska and reintroduce some tigers from Ranthambore to Saliska Tiger. So this documentary is prepared by Rajasthan Forest Department, and all credits goes to the Rajasthan Forest Department. So after screening this documentary, we can have some question and answers, and we just see how the tigers, they have been repopulated. Just a minute, I'll share my screen. It is visible.
Yes, sir.
Voice is clear?
Yes.
Sir, voice is not audible to me, I think.
Yes,
Yes,
to
sir.
me as well.
Voice is not audible.
No, sir. Sir, you need to click on share sound button, sir.
Pardon?
When you, sir, when you click on share screen button, you can see a share sound button also on bottom corner left side.
Share sound hoga, sir. Yes.
Sir, please reshare your screen, sir. I am
Reshare.
stopping. Yeah, yes, sir. I am stopping your share. And please click again share screen button.
Okay. Yes.
Now you can see share sound button.
Share sound. Yes, yes, yes.
Yes, yes. Please check.
ठीक है सर.
Ministry of Environment and Forest gave the approval of the project on 26th March 2008 and gave a go-ahead for the reintroduction of tiger. The preparation for the project was initiated simultaneously in Sariska and Ranthambore from 1st April 2008. 28 June was then targeted as the date for translocation of the first tiger from Ranthambore. The field operation commenced on 20th of June with the arrival of Director Wildlife Institute of India at Ranthambore. On the morning of 28 June, Ranthambore woke up to a heavy overcast sky and torrential rains continuing from the previous night. The teams were stationed in northern part of the park, commonly known as Dara area. As the presence of the animals was located near a kill close to Anandpura Chowki of the park, the teams were dispatched immediately to Anandpura and Dara for tranquilizing the animal. The darting of the animal was done at about 7:30 a.m., standing from a distance of about 20 meters. The dart was targeted in the hind quarter of the animal, as this was the well-muscled area. The equipment included a tele-injection using telescopic gun which was powered by air. The lightweight plastic dart of 3.5 ml capacity was filled with a mixture of xylazine and ketamine as drugs of choice for sedation. The effect of the drug was observed after 15 minutes, and immediately the team approached the animal for close observations. The tiger was radio collared. It also had a GPS and a VHF tracking system. It was programmed such that the signals were received through Argos communication satellite orbiting the Earth. The GPS of the radio collars were tuned to the signals every hour so that the mapping of the movement of animal could be accurately done on daily basis. The animal was shifted to a stretcher and placed in a lateral position, and then after moved toward the transport container designed for the purpose of airlifting. Strong and well-ventilated container for the tiger had the provision of small window for giving any medicine during transportation and was designed by using IUCN guidelines. An Indian Air Force Mi-17 helicopter was used to transport the tiger in the crate to Sariska. Dara male was 3 to 4 years old tiger and had been selected out of 5 animals of Ranthambore mainly on account of its good health and non-aggressive temperament. Its health and body measurements were based on regular observations, and they gave it the best possible candidature for translocation. The morning time of immobilization was selected mainly because of the ambient temperature of 32 degrees and favorable conditions prevailed that morning. This minimized the chances of overheating of the body due to sedation. During the flight, the animal was monitored every 5 to 10 minutes, as it took almost 40 minutes to reach the destination to Sariska. The respiration of the animal was found to be regular and deep during the entire period of operation. Tigers in India are found in diverse habitats, including dry, moist, and semi-evergreen swamps and mangrove forests. The Sariska Tiger Reserve is the westernmost distribution limit of tigers in India. The disappearance of tigers of Sariska has shown us the possibilities of the looming threat on isolated tiger populations in many parts of the country. It is important to presume that such losses and local extinction in future will be more frequent, and we will have to undertake immediate reintroduction and restoration programs to save our natural heritage. Rajasthan Forest Department, in consultation with the Wildlife Institute of India, prepared a detailed protocol and plan of operation to translocate 5 tigers from Ranthambore National Park to Sariska Sanctuary that is situated about 200 kilometers north of Ranthambore. The operation of translocation was preceded with an overall development of Sariska and Ranthambore habitats. Steps were taken to effectively sanitize the tiger reserves against poaching and minimize human and cattle disturbances. Shifting of villages in Sariska from the core area was kept as the topmost priority. All this was completed by the end of 2007. The Ministry of Environment and Forest gave the approval of the project on 26th March 2008 and gave a go-ahead for the reintroduction of tiger. The selection of the enclosure site was such that it completely matched the typical forested habitat of the tiger with a small water hole inside. A 12 feet high iron fence with a sliding gate was raised on the boundary of the enclosure. 3 observation posts were constructed at critical points in order to monitor the movement of the animal inside the enclosure. Visual barrier of yellow color cloth was wrapped around the enclosure so that the animal does not make any attempt to injure itself while trying to break the fence. It also rained in Sariska heavily on the morning of 28th June, thus delaying the landing of the helicopter by almost 3 hours. The chopper took off past 11:30 from Ranthambore and reached Sariska after 40 minutes. The container was offloaded from the chopper on a vehicle kept at the helipad. The vehicle was then taken close to the enclosure and the animal was directly released through a sliding gate. It was interesting to find the male tiger look back at us with a confused look and finding itself in the new home. The first phase of translocation was thus successfully completed in Sariska on June the 28th.
So I think I hope you have enjoyed this.
Participants, all are muted, sir. So I think, sir, we can share these questions. We have 10 questions and they have half an hour. So they will answer and they'll send back to me.
Okay.
Yes, sir. Then we will go for conclusions.
Good.
Okay, so participants, I'm sharing.
You can share this Google Doc, na?
सर वो बन नहीं पाया तो उनको मैंने एक वर्ड फाइल बना दी है, so they will just click and send it.
Can I ask something quickly?
Yes, ma'am. Sure. No problem. We have time.
Randeep ji, so what is the status of Sariska tigers now? So where are all these? How many were reintroduced in Sariska? And are all of them are alive? And how is going on in Sariska at the moment?
Initially, we have introduced 2 male and 3 females, and later on, the 5 females and 8 females.
Voice is not clear. 1 minute. Okay. Yeah, please.
5 male and 8 females, they have relocated. So few of them, they died due to some poisoning, and some suddenly they disappear. We don't know the fate of these animals. And some females, they have bred, and they have littered 2-3 cubs, and they have grown up. Now population is doing good. Approximately 20 to 25 tigers still there in the Sariska.
So 20 to 25 tigers in Sariska. And what was the founder population? Founder individuals, how many?
Founder, there are basically 8 individuals.
8 individuals. How many generations passed already?
3.
Hardly 1.
Yes. The population is already bottleneck. Ranthambore, you know that you have company Ranthambore. The Ranthambore population is already, they have a low genetic diversity. And we have reintroduced tigers from this Ranthambore itself. So you know that a lot of bottlenecks are there and the population is highly inbred. So, but population still, they are doing good. And I think 2 or 3 females are bred and some cubs, they have grown up, some they dispersed, and some they basically died due to unknown causes.
Wow, great. So you were there, right, during the reintroduction phase?
Yeah. Reintroduction, I have radio colored, all these radio colored done by me.
Great, nice, huh?
Okay, so you can share it.
Are you coming to India?
I can't hear. The voice is not clear to me.
Voice is not clear. When are you coming to India?
When I'm coming at the end of July.
10th of July. Do visit Amity?
Yes, I think this time maybe yes, with Sujit. I am planning with you guys.
Yes, you can come and plan some projects.
Yes. It's really interesting. And now I think it's getting more interesting with all these talks and people coming together on this platform. We can really do some good work actually.
So and with Ralph?
Sorry?
Ralph?
Ralph, yes. I met him last year actually. He just invited me for a talk. So I went to see him, but he's always very busy with other things. So if I ask him one thing, he will answer me a year later. So it's like that. They all get busy.
Great.
So I think we cannot share Word document through this chat, sir. I think, sir, we can wait for some time, and I'll make this Google Drive, and then I'll just do it. It will take 10 minutes only.
Sure.
Because we have time, sir. So, hello participants. I think for this evaluation, we will give question tomorrow. So tomorrow we'll share question with you all, and it will take maximum 20 minutes to answer all the questions. So it is end of today's FDP, and we had almost a total 6 presenters today. So for vote of thanks for today's FDP, I would like to invite our faculty colleague, Dr. Murali Krishna. Murali sir, please join the session and give your vote of thanks for this session.
Thank you, sir. Good evening, everyone. I'm here to give the formal vote of thanks for the faculty development program that has actually been held since yesterday. So to begin with, we had wonderful sessions since morning, and we had a lot of people who actually have come and have presented and actually enlightened us with the knowledge that they have and with the wonderful presentations. So starting with Dr. Ashutosh Singh, who is a scientist at Wildlife Institute of India, Southern Regional Center, Coimbatore, who spoke about glaciation and climate change, impacts on bird speciation and habitat invasion, cases from Himalayas. Then we had Dr. Sujit Singh, Assistant Professor from Amity Institute of Forestry and Wildlife, Amity University, Noida, who spoke on genetics and genomics, a toolbox in wildlife conservation. Then we had Professor Jouni Aspai from University of Oulu, Finland, who spoke about level of inbreeding and inbreeding depression among Finno-Scandinavian wolves. And then we have Dr. Puneet Pandey, who was our former colleague even, and currently working as senior research scientist in Seoul National University, South Korea. He spoke about protecting wildlife through technical scientific cooperation. Then followed by Dr. Rita Sharma, research affiliate from Wildlife Institute of India. She spoke about assessing the suitability of genome wild SNP markers for the conservation of endangered Borneo elephant. And then we had Dr. Bhim Dutt Joshi, scientist from Zoological Survey of India, Kolkata, who spoke about conservation genomics of endangered mammals in Indian Himalayan region. So we had wonderful set of speakers who enlightened us with the wonderful presentations. We are very thankful to all the speakers from the heart for enlightening us with the knowledge that they have. And we also thank the speakers on behalf of Amity University, Noida, even. And we also thank the participants who are here, who have been listening to all the talks with all the patients and have been asking wonderful questions and getting the feedback from the speakers. Thank you, everyone.
Thank you, Murali sir. Thank you for your vote of thanks, and thank you all for joining, all the especially participants and our speakers. And this is end of today's session. So we'll meet tomorrow with another topic of GIS, I think. So thank you all for joining, and this is now I'm ending this session. Thank you all.
Thank you.
Thank you all.